Daniel, who's already arrived. I welcome you all. Good morning to my colleagues and my dear friends and my uh, researchers. And good evening to Danielle Gilkis. She has been really she has taken out time to join us. I really welcome her. And I thank the organizers for inviting me to do the honors today. I thank Dr. Sunita Jetli, my co-chair, to join with us. So together we welcome Daniel, and the events will go like we will introduce you, and then we will take the question answers after and after that we shall have a wee bit of interaction and concluding sessions where we will further interact with you, with the faculty members may also want to interact with you, and followed with a vote of thanks. So this is going to be your 40 minutes, the next 40 minutes. With that, officially, now, I'd like to give the brief sketch of Danielle Gilkis. She earned her Bachelor's of Sciences in Chemical Engineering and Master's of Science in Material Science from the University of Florida. After a brief time in the industry, she pursued her PhD in cancer biology from the University of South Florida. Her postdoctoral fellowship was conducted at Johns Hopkins School of Medicine, where she was mentored by the Nobel laureate, Professor Greg Semenza. She is currently an assistant professor of oncology at the Johns Hopkins University School of Medicine in the Department of Oncology, with a courtesy appointment in the Department of Chemical and Biomolecular Engineering. The overall objective of her laboratory is to identify biomarkers and therapeutic strategies that focus on prolonging survival and preventing and treating metastasis for breast cancer patients. Her work has focused on the role of hypoxia and hypoxia-inducible factors in promoting breast cancer metastasis by promoting changes in the structure and composition of the extracellular matrix, mainly through the collagen matrix modifications. Daniel has identified many HIF-inducible target genes that are required for the metastasis in animal models. What is interesting is her current work attempts to determine whether hypoxia-inducible genes are transiently expressed and serve to initiate the metastatic cascade, or whether they chronically contribute to the metastatic process. To this end, her lab has developed many models, including a transgenic mouse model to fit the MAP cells, allowing hypoxic cells to be tracked to distant organs. It is an honor and a pleasure to invite her to enlighten the audience on the topic chosen by her, fate mapping hypoxic cells during metastasis. With this, I welcome Daniel. The floor is yours.
That was just such a beautiful introduction. The honor is all mine. I thank you so much for that. I'm hopefully sharing my screen with you.
Yes, you are.
Okay,
yes.
Wonderful. Step one. Okay. So again, today I'm going to be talking about the role of hypoxia in cancer metastasis and the model that we've generated. So thank you so much to the organizers for inviting me. I'm going to first just start out talking about what is hypoxia. So everyone on this call, we're all breathing in air, which contains 21% oxygen. And in order for the oxygen to get into our tissue, it must diffuse from the alveoli in our lung spaces into our bloodstream. So that's what we're showing here. And this happens with red blood cells, which carry oxygen to all the organs in our body. So the oxygen levels in our peripheral organs can range anywhere between 3 and 7.5%. And that just depends upon the organ that we're considering. And we call a tissue hypoxic when the oxygen levels drop below the normal oxygen level for any particular organ. So in tumors, what does this look like? Sorry, I'm moving things around. There we go. So this is a little cartoon of a tumor. So basically, tumor hypoxia results from multiple mechanisms. So first, you can have abnormalities in tumor blood vessels and structure. That's what we're showing here in this little picture. This can lead to a decrease in the oxygen delivery, and that's relative to normal tissue. So what you have is this relatively high rate of tumor cell proliferation as the tumor progresses and continues to grow. So you have this increase in oxygen consumption by the tumor tissue. And the net result of this is a decrease in oxygen supply, but an increase in oxygen demand. And so we call this hypoxia. And we know that this can occur over a very short length scale. So here we're showing just 1/10 of a millimeter where hypoxia can develop. Here I'm showing a full cross-section of a mouse mammary tumor that we've stained with hypoxy probe, and that's in brown, and that's to mark the hypoxic cells. So what you can appreciate is that these hypoxic cells line the necrotic core, this white tissue here, which is essentially dead tissue. Then if we look a little bit closer, this inset is showing you cancer cells that are surrounding two blood vessels. So here's the two blood vessels, and the cancer cells are eventually hypoxic when they become at a distance maybe greater than about 200 microns from the blood vessel. So that's the brown staining here. And so just to summarize that, you have a nice blood vessel, you have oxygenated cells that are surrounding the blood vessel. At a certain distance, hypoxia begins to occur, a decrease in oxygen, and then necrosis, or actually the cells become so starved of oxygen that they die. So there have been groundbreaking clinical studies in which oxygen measurements of breast cancer have been obtained in situ using these sort of needle-type microelectrodes. And these studies have been conducted in humans. And what I'm showing you here is a histogram of oxygen concentrations. And this is one of the studies. In this study, there were measurements taken at over 1,000 points within the normal tissue of 16 participants. And the median partial pressure of oxygen within this normal breast tissue is 65 millimeters of mercury. You can equate that to about 8.6% oxygen concentrations. Then if we look at breast cancer tissue, I hope you can appreciate that the median oxygen concentration levels drop, and they actually drop below 1.5%. And that's what's shown here in this graph, where we have oxygen concentration versus the points that were measured. So there have been remarkable similar results that have been obtained. So if we look at meta-analysis that have involved more than 700 patients that have either cervical cancer or 500 patients with head and neck cancer, basically any cancer that we can measure with a needle-type electrode have been measured for hypoxia. And in these cancers, multiple studies have demonstrated an association between oxygen concentrations that are lower than 1% and decreased disease-free survival. So what happens under these hypoxic conditions? Well, through the years, we've learned that hypoxia-inducible factors are well recognized as the master regulators of hypoxia. So how does this regulation occur? Well, there are these specific hydroxylase sites on these HIF proteins, and these proline residues that basically reside in conserved core domains of both HIF-1 alpha and HIF-2 alpha. They are subjected to oxygen-dependent hydroxylation. So when oxygen is around, you have hydroxylation of these sites by the prolyl-4 hydroxylase domain proteins. The hydroxylation event then allows the HIF-1 alpha or HIF-2 alpha to interact with a von Hippel-Lindau or VHL tumor suppressor protein. And this protein is a substrate-specific component of the E3 ubiquitin ligase. And so when this substrate binds here, what happens is HIF-1 alpha and HIF-2 or HIF-2 alpha are ubiquitinated, and then they're degraded through the proteasome. But what happens under oxygen conditions, when we get rid of the oxygen, these enzymes become inactive, these prolyl hydroxylases, because they no longer have oxygen as a substrate. And this leads to HIF-1 alpha stabilization, accumulation, dimerization with HIF-1 beta, translocation to the nucleus. Here within the nucleus, the HIFs can bind to their target gene sequences that contain hypoxia responsive elements, and they activate the transcription of the target genes. And we know that more than 200 genes have been shown to be HIF targets. And these genes involve metabolic reprogramming, erythropoiesis, angiogenesis, and many more. So if you can think about a normal tissue that's injured, the response is actually the response of HIF-1 alpha being induced is really a healthy response, because there will be more red blood cells that are produced, there'll be more blood vessels that are made, and all of this is in an effort to restore oxygen balance. But if we think about cancers, on the other hand, in a cancer, this can lead to adaptations that allow those cells to survive when they should be really starved of oxygen, and they should die. So recently, our lab was interested in establishing a hypoxic signature, and we did this by comparing the differential gene expression in 32 different breast cancer cell lines that we are either exposed to 20% oxygen. So this is when HIF1 alphas are very low, or 1% oxygen. And this is when HIF1 alpha and HIF1 alpha and HIF2 alpha rise to very high levels. And what we showed, which was very interesting, is while there were more than 3,000 genes that were differentially expressed in each of the individual cell lines, only 42 genes shared a common differential expression. And so we went on to use this consensus sequence to look at patients, and we stratified them based on this 42 gene signature that we found. And we found that this 42 genes signature set was prognostic for overall survival. And this was done with retrospective data. But similar to our studies, there have been many other studies that have been conducted either with their own different hypoxic signatures or looking at HIF1 alpha or HIF2 alpha by staining for immunohistochemistry, or using immunohistochemistry to stain for downstream target genes. So again, there are many different hypoxia-inducible gene lists that you'll find. Unfortunately for our field, no one gene set or biomarker has been agreed upon. Really, at present, there's no biomarker that's used clinically for any type of decision-making. So in our lab, we wanted to ask a simple question, and that was, we wanted to ask if the survival mechanisms that keep these normal organs healthy can be sabotaged by cancer cells, and that they can be continued to be used to escape the primary tumor and metastasize to regions that are not hypoxic, such as adjacent normal tissue or different organs. So the way that we went about doing this was we used a lentiviral delivery approach. So we generated a dual vector hypoxia fate mapping system. So the first vector that I'm showing you here allows for the expression of a red fluorescent protein, in this case DS-red, to be expressed. And it has a stop codon that's flanked by tandem LOXP sites that precede the gene encoding a green fluorescent protein. And then we generated a second vector. And this second vector contains an altered Cree gene that's been modified with the addition of basically an oxygen-dependent degradation domain, but that's also under the transcriptional control of a HIF DNA binding sequence. So basically what occurs is under low oxygen conditions, HIF1 alpha and HIF2 alpha are expressed. They bind to the promoter, activating Cree expression, which acts as a scissor to delete out the DS-red gene, and the cell begins to express GFP protein. And so I kind of think of this as if everybody in this room were wearing a red shirt and we had this tightrope walking competition. So if you were successful at crossing the tightrope, that would mean that you were successful, say, at surviving under hypoxic conditions. Then you would get this green shirt to wear. So we could now follow you everywhere around the room. And for the tumor, this means that we can monitor a hypoxic cell even when it leaves the hypoxic microenvironment and becomes reoxygenated. So what does this look like in cells? So these are MDA-MB231 metastatic breast cancer cells. We've exposed them to 20% oxygen conditions or 5% oxygen conditions, which is closer to what we'd find in normal breast tissue, or we've exposed them to 0.5% oxygen conditions. And you can see that they're green here. And so in real time, hopefully I can show this movie. So at time 0.0, we've exposed the cells to 0.5% oxygen, and then we're following them over time to watch the fluorescent change from DS-red to GFP. We can also use these same cells to generate 3D models. So we've looked at MCF7 cells that we've also employed this fate mapping system into. And you can see that at time 0, these spheroids are completely red. And then at the end point of the experiment at 14 days, you can see the existence of these GFP expressing cells within the center of the tumor. And in the MDA-MB231 cells, these cells when they're placed into a matrigel matrix, into a collagen matrix, they really like to spread out as opposed to the MCF7 cells. But you can see, hopefully you can appreciate this hypoxic core that's developing. And if we look at this in 3D, you can see that the GFP cells, we can find them. They can actually migrate to the edge of the tumor spheroid, but they are mainly localized in the center of the spheroid. We can also directly measure using an optical electrode the oxygen concentrations within the center of the spheroid, which we find to be on the order of 1% compared to in the collagen matrix itself, which appears to be closer to about 12.5%. So next, we're really interested in planting these cancer cells into the mammary fat pad of immunodeficient mice so we could see how the tumors progress over time. So what you can see here is that when the tumor is quite small, all of the cells are expressing DS-RED. Then as they grow larger, you can see the existence of this necrotic core of dead tissue and then surrounding GFP positive cells surrounding this necrotic core. But we were really surprised to find that the population of GFP positive cells, or those cells that were exposed to hypoxia and had permanently changed to green, increased over time. And we can find them invading into the red regions of the tumor. So here we decided to chat with our colleagues and tried to come up with a mathematical model to help us to inform how this invasion can actually occur. So to inform the model, we first assessed the spatial distribution of oxygen in our MDA-MB derived orthotopic tumors. And we did this using an optical detection probe. And we attached this to a micrometer so that we could actually record the oxygen concentrations at 0.5 micron depths within the viable rim of the tumor. So closest to the necrotic core, the oxygen concentrations are closer to 1%. And then as we retract this needle-based probe to the edge of the tumor or the periphery of the tissue is on the order of about 6% oxygen. So this suggested to us that this was really simulating what was occurring in human breast cancer, at least the oxygen concentrations that we have. Next, we isolated green and red cells from the primary tumor that I just showed you, and we used them to generate spheroids. And what we did so that we could monitor the migration of individual cells within a spheroid was we mixed 10 to 20% of our tumor-derived cells that were labeled with green or red with 80 to 90% of unlabeled naive MDA231 cells. And we did this so that we could visualize the green and red cells as they migrated within the spheroid. And we used live cell imaging here. And we tracked the individual cells invading into the matrix. And we could come up with a measurement for their average speed and for their maximum displacement. So using this data, we work with our collaborators at Indiana University, who are in the Department of Intelligence Systems, who used the data to kind of tune a computational model to see if we could match our observations. So the lab considered three important parameters. The first parameter was the length of time, we called it phenotypic persistence, was the length of time a green cell has that it can leave the hypoxic region, but also maintain a migratory phenotype. So you can sort of think of this as like a fight or flight feeling of panic that a cell may have after it's recovered from a lapse in oxygen concentration. Next, we considered how biased a cell would be towards wanting to get to the region of higher oxygen concentrations. So the Macklin labs simulated a tumor that starts off with all red cells here, and they set the persistence time or the panic time to 0. And what they found was that you can see that the randomness of migration alone by setting the persistence time to 0 wasn't sufficient to drive the invasion that we were actually seeing in our experiments. So next they ramped up this sort of memory of being hypoxic to 50 hours. And here you can now see that there was the invasion of these green cells into the red cell region. And the last test that they performed was to see what would happen if the cells were permanently panicked. So they just maintain this constant migratory phenotype. And if we did that, that allowed these cells to rapidly escape the tumor, which we did not see experimentally. So we didn't think that this was true. Lastly, our observational study, when we did it, we noticed that there was a large heterogeneity in the number of GFP cells that became more migratory upon hypoxia. So they all weren't reacting the same way. So you could see that we had a very large standard deviation. So to address this observation, the Macklin Lab next introduced a percent responders term. So this considered how many cells would actually respond to the hypoxic stimulus that they were given. And that's finally shown here in this model. So we have a bias towards oxygen. We have a persistence time towards remembering that hypoxic stimulus, and then we have a percentage of cells that can respond to the stress. And so this mathematical model actually looks like this when you perform the agent-based model. So you see that all of the cells are red. Some of them are responding to hypoxia and turning green. Then there's going to be blue cells, and these are the necrotic cells or dead cells that form. And this allowed us to really have a dynamic and evolving view of how hypoxic cells were responding in the tumor, based on, as compared to our animal models, where really I could only show you static pictures. And so when we compared the computational model that's shown here with our observational model with the tumor itself shown here, we observed really well-defined, persistent green invasive plumes here were recapitulated in the mathematical model. And we could further go into 3D, and here's a 3D look at the model. But then on the right-hand side, we have a portion of a 3D tumor that we've used cubic clearing methods to clear the tumor. And then we've imaged them on a confocal microscope using multiple Z-stacks to reconstruct the tumor. And that's shown here. And the bottom here, we're showing a surface rendering of the 3D construction that also you can appreciate the presence of these green plumes that are invading into the red region. Okay, but really, what about metastasis? That was sort of our original focus. So stepping back to metastasis. So the way that we set up our model was we developed orthotopic tumors in our recipient mice, and then we removed the tumors at 20 days after they had formed. And so at the 20-day time point, what we found was that 20% of the cells were GFP positive or had experienced hypoxia in the tumor, and 80% were red, basically, or DS red expressing. But then if we looked at the lungs, what you can see here is a whole mount imaging, fluorescent imaging of the lung for green and red, and then a single tissue section of the green and red imaging. And what we found is that while only 20% of the tumors at the time of removal had GFP positive cells, closer to 50% of the cells in the lung had GFP positive cells. So if we divided these two, the available cells versus the cells that wound up in the lung, we found that we had a probability of GFP cells had a 5 times increased probability to be able to metastasize to the lung. And what was perhaps even more interesting was that these GFP positive cells were actually 25 times more potent at forming brain metastases. And so this is a whole brain shown here, and then analyzed, the metastatic burden was analyzed by flow cytometry here. And you can see in the pie chart that the majority of the cells are GFP expressing that we find in the brain. We can then go on to optically clear the brain's sections. And so here I'm showing you where we've cleared the brain, and then we've performed immunofluorescent labeling of CD31, and that's the purple, and that's so that we could see the blood vessels in the brain. And then the green cells here are the cells that experienced hypoxia in the primary tumor and that had mated or metastasized to the brain. And further, when we go to a higher magnification, we can actually see that the GFP positive cells are able to extravasate from the blood vessels into the brain to form a metastatic lesion, and they're able to survive within the brain microenvironment. The next thing that we wanted to do was use another technology where we can actually map the entire brain of the mouse, and then we can map the brain and the GFP positive regions onto a reference atlas of the mouse brain. And when we do that, you can see that the majority of the cancer cells that we find are within the brain ventricular system. And this is a common site of leptomeningeal metastasis that occurs in humans. Next, we found that the cerebellum and the cerebral cortex were the portions of the brain parenchyma, which had the next highest incidence of metastasis. And this also occurs as a site in humans that have the highest incidence of metastasis. So we see that these cancer cells that are exposed to hypoxia in the primary tumor are better at metastasizing. So we wanted to test this in a second model. So what we did was we generated a transgenic mouse. And so our mouse here is the oxygen responsive mouse that's expressing Cre only under hypoxic conditions. And we bred this mouse to a Cre reporter mouse. And this Cre reporter mouse is similar to the original vector that I showed. So the mice are all red. All the organs of the mouse are red. But when Cre is expressed, they turn green. So we then bred our double transgenic mouse here to the MMTV PYMT mouse model of breast cancer that's driven by the PYMT oncogene. And this allowed us to generate a triple transgenic mouse. So this triple transgenic mouse will spontaneously develop breast cancer, and then the cells that are hypoxic within the breast cancer will then turn GFP expressing. And so this mouse model of the PYMT model has already been shown to be a nice model for following the steps of progression of breast cancer. So from hyperplasia to DCIS to early carcinoma, and later on to invasive carcinoma. And so what we've done is just simply layer on the ability to look at cells that are hypoxic within this tumor. So in the hyperplasia stage, we really don't see cells that are hypoxic. I'll show you higher magnifications of DCIS in a moment. In early carcinoma, we can see the beginning of GFP positive cells, and then we can see many more hypoxia exposed cells within these invasive carcinomas. And here's just showing you the DCIS lesion, which requires a higher magnification to begin to see the presence of RFP, or the red fluorescent protein, changing to GFP fluorescent. So just to show you that our model is actually working and changing under hypoxic conditions, we excised the tumors from these mice, and then we formed organoids in vitro culture. And the organoids started at various sizes, and then they grew to various sizes. And hopefully you can see that the red color within the tumors eventually changes such that a portion of the cells are expressing GFP. And it may be more beneficial to see the organoid here, where you see the organoid growing and then becoming GFP positive. But what happens to metastasis? So in our transgenic model, similar to the model where we used immunodeficient mice, here we're using immune competent mice. And again, we find that these mice have a 4 times increase in the GFP positive cells to metastasize to the lung. We then transplanted these into a secondary model. And in the secondary model, the mice were immunodeficient. And we just took the organoids from the primary transgenic model and put them into the NSG model. And again, you can see this increase in metastatic potency. Okay, but why? So we decided that we needed to figure out which step in the metastatic cascade these cells had a better preference for, were able to overcome. So we looked at the whole metastatic cascade. And in the beginning, I showed you some motility data that we did see that hypoxia exposed cells had a little bit of an increase in their ability to locally invade. But next we wanted to assess intravasation, because we know that circulating tumor cells, they're shed each day, but that metastasis is really an inefficient process. So we hypothesized that hypoxic exposure could have potentially a role in intravasation. So to test intravasation, or the ability of a cell to get in from the primary tumor into the bloodstream, we assess the number of circulating tumor cells in the whole blood. So just like the lung, When we look at the number of CTCs, we find that GFP cells were 4 times more likely to be found in the bloodstream, and that the GFP cells were less apoptotic than the DS-red cells. So this sort of led us to hypothesize that the cells exposed to hypoxia may be primed to survive stresses in the bloodstream. And we know that hypoxia exposure from many prior works prior to ours have shown that hypoxia exposure can allow the cell to resist ROS. And we know that ROS, reactive oxygen species can occur in the bloodstream. So we next had to determine whether the blood cells were better at getting actually into the bloodstream or better at surviving. And so in order to question or to answer this question, we questioned whether the cells were more resistant to oxidative stress that they would encounter in the bloodstream. So in order to evaluate the mitochondrial ROS levels directly, we isolated the GFP positive and the DS-res positive cancer cells directly from the blood in the mice, and then we stained them for mito-ROS. And then we performed a similar experiment, but here we treated the freshly harvested circulating tumor cells with the ROS inhibitor and acetylcysteine or NAC. And what we found in our results is that while the ROS levels in the DS-red CTCs were reduced to the ROS levels measured in GFP positive, were reduced to the levels of GFP positive cells, but we did not find changes when we treated the cells with NAC. Likewise, to further explore this ROS-resistant phenotype, we treated GFP positive or DS-red positive cells that we derived directly from the primary tumor here. We treated them with hydrogen peroxide for about 1 hour. And this was to sort of promote ROS induction or ROS-induced apoptosis. And then we evaluated the survival of the cells 48 hours later. So these are the cells treated with 0 micromolar of the hydrogen peroxide, 50, and then 500. And so what we found is that the GFP positive cells were significantly more resistant to the ROS insult of hydrogen peroxide than the DS-red positive cells. And this is just a quantification of the experiment to show that we measured multiple different areas and multiple wells for this experiment. So lastly, we wanted to try to understand why these GFP positive cells have an enhanced ability to metastasize. So to look at this, we performed a similar experiment to the one that I showed you initially, where we removed the primary tumors at 20 days, and then we used flow sorting to derive DS-red positive and GFP positive populations from the primary tumor. Then we waited for lung metastasis to occur and did the same thing at that time point. We collected the different subpopulations, and then we submitted them to RNA sequencing. So our first finding is that we first compare the gene expression changes caused by intratumoral hypoxia. So that is the difference between the GFP expressing cells in the tumor and the DS-red expressing in the tumor. And we also compared cell culture. So what we find when we expose the cells in our laboratory to 1% oxygen conditions for 24 hours or to 20% oxygen conditions. And we were really surprised that only 41 RNA transcripts were regulated in common between these GFP positive cells versus these DS-red positive cells versus what we're used to when we do our hypoxia experiments in vitro in the laboratory. So this really told us that our experimental hypoxia conditions don't entirely recapitulate what we're finding in vivo. And I'll skip through this part so that I can cover the next part. The next part is we wanted to look at the pathways that were most enriched in the GFP positive cells, both in the primary tumor and in the lung. And what we found is that NF-kappa B was the most regulated pathway in these GFP positive cells. And we know that NF-kappa B has been implicated in oxidative stress. So right now our lab has been focused on trying to understand this paradigm and to see if NF-kappa B upregulation under hypoxic conditions may be a ROS protective mechanism for these GFP positive cells. So I really only have a couple preliminary experiments to address this question. So in order to do this, we incorporated a third vector into our system, which we borrowed from this science paper. And this third vector expresses MCATE, which is a far red fluorescent protein. And this occurs on NF-kappa B binding. So now we're able to observe both hypoxia and NF-kappa B activity. And so what we found is that in the GFP positive cells, NF-kappa B expression is higher in the tumor, in the GFP cells in the tumor, the blood, as well as the lung. And we could, in addition to flow cytometry, which was done here, we could also do some imaging of the lungs of the mice and look at the GFP positive regions, which are co-regulated or co-localized, with MCATE expressive regions, whereas the DS-RED positive regions are not. And so we are now have ongoing experiments in the lab to look further about what exactly NF-kappa B is doing in our system. So basically, what we found within our study is that the GFP positive cells appear to have a 5 times increase in their ability to survive within the bloodstream. And this is allowing them to be more potent in their ability to metastasize. And this is at least for the lung. We haven't done, we're initiating experiments now to try to understand brain metastases and our findings within that setting. So in summary, hypoxia occurs in tumors, and this is due to an imbalance between oxygen supply and oxygen demand. Hypoxia, what we know, promotes HIF protein expression, and that leads to aberrant gene expression patterns. We were able to develop a hypoxia fate mapping strategy, and we can do this with mouse models as well as in cell lines. We found that hypoxic cells, at least in our animal models, are 5 times more likely to form a lung metastasis than cells that have never been exposed to hypoxia in the primary tumor. And then we have this interesting finding that they have a preference for the brain over the lung, but we really don't have any understanding as to why this might be. We know that cells that are exposed to hypoxia in the primary tumor are poised to really tolerate and survive the oxidative stress that occurs within the bloodstream when the cells are reoxygenated. And tracking these cells that become hypoxic in the primary tumor en route has revealed this phenotype that's protective against ROS. And now we're really evaluating the role of NF-kappa B and to see if it has any potential protection from ROS, and that whether or not it can be implicated in the findings that we have. So I'd like to thank the members of the laboratory that are here that contributed to all of this work. Of course, the many collaborators across Hopkins and beyond, and then some of our funding sources. And then I really want to thank each and every one of you for listening to this talk today. And I appreciate your attendance. And I'm happy to take any questions.
Thank you so much. Thank you so much. I think so now the dais is open for your questions. And there'll be numerous, I know, because this is a very innovative concept she's come up with. And post-COVID, hypoxia is the clutch word, you know. So,
we haven't gotten into any COVID research. We've stuck with our cancer research. But so shall I look at the chat?
You can type in your questions. I think so few have come up, Dr. Gilkis. Can we stop these cells to become metastatic cancer? This is one of the questions.
So we would like to figure out if NF-kappa B is responsible, and if so, we're going to try to do CRISPR deletion studies to see if that alters their ability to survive better in the bloodstream or not. So to see really if that is enhancing their ability to survive against reactive oxygen species. That's what we're testing. And there are some NF-kappa B inhibitors that we're testing in animal models.
Indranil Chattopadhyay wants to ask, is there any relation between hypoxia and cytokine storm and mitochondrial dysfunction?
I'm sure that there definitely is. Our lab isn't particularly looking at the cytokine storm, but I think we could definitely use our method for marking these cells to be able to kind of either with staining or pulling the cells out of the tumor to ask questions about the cytokines that are being released, either by the microenvironment, the cells that are exposed to hypoxia in the microenvironment, or by the cancer cells themselves. That would be a really interesting study. We'd also like to find an immunologist that would want to help us, because I think in our transgenic model, we have the opportunity to really look at the immune cells, but we don't in our lab have specific background in that area. So
another one has popped up from Himanshi Prakash Meena. What should be the normal level of oxygen to prevent hypoxia?
So that's a really
Very
excellent, that's an excellent question, because it really depends upon the organ site. So we know, like when we're breathing the air into our lungs, the partial pressure of oxygen is going to drop as we're breathing in the air, and then it hits the alveolar spaces. So the air has 21%, but in our lungs, we're sitting closer to like 13 to 15%. So that's the normal oxygen concentration. So maybe for the lung, you could consider 8% almost being hypoxic. Whereas in a different organ, such as the breast, we know that 8% is more of a normal concentration. And so then like 1% is going to be hypoxic. I will comment that 1% oxygen at 1% oxygen levels, that's the where the half maximal induction of HIF1 alpha is. So that's kind of why we use 1%. That's where you have the half maximal induction of HIF. You don't have super huge levels, but not super low levels.
Is ROS more potent in hypoxia or normoxia? Misha Lamichene has asked.
So in studies that have not been conducted by our lab, but that are in the literature, HIF1 alpha in particular has been known to, HIF1 alpha, and I should say HIF2 alpha to an extent, has been known to upregulate suppressors of ROS. So I think, prior work to ours has already begun to show that a cell that's been exposed to hypoxia sort of is already expressing these genes, such as BNIP3 and others that can help to survive a ROS insult. I hope that answers it.
I think so. Professor Kamra wants to ask the question. I think so. Ma'am, please unmute yourself.
Yeah, definitely. It was a really exciting talk. Very, very exciting. It's really got our antenna alert that we could do a lot of work like this. A little thing I want to know is, can we actually correlate this with chemoresistance too?
Okay.
Have a metastasis, we have a chemoresistance. Can we do your work or get an idea when we are doing the fate mapping?
So yeah, that's a great question. We just published a few kind of preliminary results where we looked at chemoresistance. So we did I think what you're suggesting is we treated mice after we removed the primary tumor with common chemotherapeutic agents that are used in the clinic to determine whether these GFP positive cells are more chemoresistant. Yes. So that was definitely on our radar.
I have done a little work on that. So I just thought that, is there another group looking into chemoresistance?
So definitely there are other groups that are looking at chemoresistance, and in fact, like the Semenza lab is looking at chemoresistance even from the aspect that drugs like paclitaxel seem to be inducing HIF1 alpha. So it's very interesting that these kind of things can occur, and I think there's a lot of mechanisms under hypoxic conditions, not just HIF.
There's a lot of crosstalk, and also when you're looking at the extracellular matrix, which is the most important thing, it is a trigger to metastasis. It is not for supporting; it is actually supporting metastasis. And that is the time when we are looking into different proteins. So have you got any idea whether desmoglein 2 was also looked into for hypoxic controls? Because we have to see what is pushing the cell away, which are the adhesion molecules which are being moved away. Then only it can go out of the solid tumor.
I agree with you completely on the extracellular matrix part. And I think like we overlook that part in the study. We really focus on the cancer cell with the system here. But I 100% agree with you that that's important. And I would be interested in going back to our studies on the GFP versus RFP cells. But that's going to be again in the cancer cells. I think I would do better to look at the stromal cells in the microenvironment, perhaps in that transgenic model. And that's something that I think that's really something that would be interesting to go and look at. And we have a tool there to do that, but we haven't. So I'd love to talk with you offline. You know,
of course we would. That would be very interesting.
Think about how we could do. Think about how we could do that.
Yeah, and I would like Dr. Sunita to officially give you a vote of thanks. We can't let you go. It's been so interesting. It's really been nice. Thank you.
I will begin
Thank you so much, Dr.
Because I was my basic research is also in CML, that is chronic myeloid leukemia. And we are trying to figure out a very cost-effective method for detection, which is even cheaper than a qPCR for especially the poor people, you know, based on a beacon. So when I started doing this cancer research, you know, I thought a cancer cell is like something, you know, going on a rampage. No more of multicellularity. Let me go back to my old age ethics of single cell, you know, and I want to live alone. It is like the cycle coming to complete, you know, eukaryotes and back to prokaryote. Yeah. So the organ system seems to be falling apart and the cell simply goes on a rampage thrill in the body. And there are many aspects, you know, many aspects to it, not only hypoxia, and there's so many other aspects, you know, with in vitro and in vivo differences, which you have very rightly pointed out, you know, it's like shaking your knees because what you discover in vitro doesn't at times apply in vivo. So on that backdrop, हमारे हिंदी में ना कहते हैं सार गर्भित आलेख, मतलब आपने बहुत अच्छा बोला. You gave a very lucid, clear expression. I'm just trying to introduce a bit of Hindi because you are addressing the Indian audience here. And, you know, the words will never come up to sum up the research behind this short one, you know, very short lecture which you have given. The amount of work which goes beyond it is like, you know, you cannot sum up in words. I know, single experiment going wrong 10 times. PCR simple band not coming. I mean, things like this, you know, they keep shaking your knees. So I know what you have, the exposition and the lucid and the clarity with which you simply flawlessly went over it. Amount of hard work is appreciated. And I think so the audience must have repleted themselves with a lot of concepts which you gave, you know, which textbook don't give you right now. And on that backdrop, I really want to thank, first of all, Manisha for calling me here and giving me this opportunity to be with Dr. Anita Kamra. She's just now been awarded with one of the most acclaimed awards of Delhi University. I must tell you, Daniel, about that. And I want to thank you, Daniel, because you people are really taking out time. These are precious moments, you know, you might be doing one experiment or might be discussing things in lab and coming and speaking at this hour, early morning. This is called as Suprabhat here. It's morning here. In morning we say सुप्रभात, मतलब बहुत अच्छी आपकी morning हो और आपका दिन जो हो, दिन मतलब day, शुभ हो. So on that backdrop, I first salute the divinity within you. In Hindi we say, हम आपके अंदर की ज्योति पुंज को नमस्कार करते हैं, the soul within you. And यहाँ पर बैठे जितने भी प्रोफेसर्स हैं, डॉक्टर्स हैं, फैकल्टी हैं, स्टूडेंट्स हैं, उन सब की ज्योति पुंजों को हम नमस्कार करते हैं, जो हमारे आभासिक प्रकोष्ठ, आभासिक means virtual और प्रकोष्ठ means this virtual classroom which you are sitting, हम उनका भी बहुत बहुत धन्यवाद करते हैं कि उन्होंने अपना सुबह का टाइम निकाला. इंडियन सोसाइटी में सुबह का टाइम निकालना is a very important event because breakfast and lunch and everything has to be prepared and you have to rush to your work. You must be appreciating that. So I really salute the college for organizing such a flawless, there was no disturbance, nothing, program, and all the organizers and the team behind it, and that backdrop. Namaskar once again.
Thank you. Thank you so much, everybody. Thank you. It was, and there are 206 participants right now.
Yes, yes.
Okay.
206
I think that
participants in the
calls for a
morning.
big applause.
That's
really
amazing.
great. Thank you
That's
so much,
amazing, yes.
Daniel. It was such
Yeah.
a beautiful talk. It was a very good research you've projected. Thank you,
And
ma'am.
Daniel, you were a star.
Thank you, Daniel. Thank you, Sunita. Thank you so much.
Thank you too. I hope to see you in person at some point.
Yes, yes.
That would be wonderful.
Thank you.
Thank you. Take care.
And Daniel, as a token of appreciation, this is because
Yes.
you are not charging anything from us, and everything is all John Hopkins is just giving their lectures for free without any pie. So it's a big applause, and thank you so very much.
Thank you.
Token of
Thank you.
appreciation, we are offering you a certificate. Thank you.
Thank you. That's awesome. Thank you so much.
Yeah,
That's beautiful.
yeah, yeah.
Thank you so much.
Thank you, Manisha. And
Thank you, ma'am.
I
Thank
I
you,
think you can now continue with the next speaker.
ma'am.
Yeah.
Thank you so much.
Thank
Thank
you
you so
for
much.
making
sir.
me the
Thank you.
chair.
Have a wonderful day.
Thank you, Daniel.
Thank
We'll
you.
get in
And
touch with you again.
have a peaceful night. Your sleep
Yes.
is over.
Yes,
Thank you,
so much.
Sunita, for being there. We are
Daniel,
as
Shubh
a
Ratri.
co-chair. And thank
Have
you
a
very
nice
much.
night.
Thank you, Sunita. Thank you so
Welcome.
much. Thank you, Sunita. Thank you. So we are just about to start with another speaker, and
Actually,
uh,
tea break is mentioned in the program schedule.
yes. So after
Session
this
would be
tea break,
start at 9.
9.
10,
No, we'll be
10:20.
starting at 10:20
Yeah.
with a speaker from Michigan University, and his topic of talk is also very good. It's on precision oncology in the age of interactive and integrative genomics. So it's going to be a very wonderful talk. You can just get back to your classes, students, if you're there, and the teachers. I'm really thankful for you all. And we'll start at 10:20. It's going to be a very wonderful talk. And for tomorrow, I just want to tell you all that tomorrow morning, 9 o'clock, please make your presence. Okay, the speaker has joined. Okay, we'll just start off then. The speaker has joined. And tomorrow also in the morning, we again have a speaker from John Hopkins University. She's also a very senior researcher. You will be getting to know a very a good research. You will get to know the work done. And today, Dr. In fact, just going to be a professor, Chandan Kumar Sinha. He has a lot of good work, and he's a very famous scientist from Michigan University. So let's start off. I'll be seeing the chair and the co-chair for the session, Dr. Archana from Hansraj and Dr. Purohit from the Cluster Innovation Center. We'll just introduce them.
Ma'am, actually, they have not joined yet
They have not joined.
because time is given them 10:20. So they will join according to the
Okay,
time.
then just a minute. Can you please call Dr. Archana, and can you please call Dr. Purohit also? Hello, Chandan. It's nice to see you. How are you?
Good, how are you?
Very fine, thank you. Seeing you here like this, because in our group we see each other, but we definitely talk to each other, but we don't, get to see these video calls and all. We seldom do them.
Yes.
So I think last time when we were on video call was around some months back. I think when all of us were there, the gang of
I
the
know.
college.
Yes, that was fun.
So it's so nice. But how were you able to take out time? Because you were
Yeah.
busy with some. I'm very surprised.
No, I finished that presentation.
Okay.
Hmm.
So actually that was the reason why I told these both the chairs and co-chairs to join a bit late. Maybe you will get late because you were on to a very important work. So Welcome.
It's good to see you.
Same here.
Hmm.
So,
It was a nice presentation. The previous one, I attended some of it.
acha, you were able to attend? I thought,
Mhm.
because, yesterday when I talked to you, I
Mhm.
thought you were going to be very busy. I thought maybe 10:20 also you'll not be able to make it. But
No.
it's really nice that you join and your
Mhm.
work, because we have many participants right now. I think the number is 205. And I really want people to know your work. You are doing wonderful work. So I just wanted all my students also and the teachers also to see your work. So
Yeah, that would be
you're a
great.
high five person now, a big man with
Hmm.
many publications and patents also to your credit. And of course, the chair and co-chair are going to introduce you. So that's nice.
All right. I guess we'll wait for some
We'll
more
just
time
wait
then.
because, I only told them maybe you are not able to join early. So I thought that maybe I'll tell them to be a bit late from the schedule.
That's all right.
It's what time at your place now?
It's 11:30.
11:30. Oh my God. Thank you. You only a friend can do this. No one else.
Correct.
Only a friend can do this at 11:30, and then you'll be getting free at 12:30. Oh my God. And you're an early sleeper, I know. You sleep by 10:15, I think.
It's
You are
all
a
right.
true friend, I must say. You are a true friend. Thank you, Chandan. Just a minute.
Mhm.
Will you allow me to make a call to the
Oh, yeah,
chair and the
sure. Go
coach?
ahead. Go ahead. Go
Just
ahead.
a minute. Thank
Hmm.
you. Thank you. Okay, so Chandan, so I'm only going to introduce you. Dr. Purohit from Cluster Innovation Center and Dr. Archana, we are just trying to get in touch with them. So by the time they come and give you a vote of thanks, I'll just introduce you as a speaker. So Dr. Chandan Kumar Sinha is an associate research scientist at UMICH, University of Michigan. So he has done work on precision oncology based on integrative high throughput resequencing data analysis, multi-omic analysis of cancer cohorts, the functional genomic characterization of novel cancer drivers. Sir has got 14 years, more than 14 years of experience, and he is associated with this university for quite a long time. And he has a lot of work on cancer genomics and bioinformatic research institute. Also, he's worked in 2007 to 2008. He was in the Bioinformatics Research Institute, and he did his post-graduation from Indian Institute of Science. He earned his PhD in molecular biology in 92 to 1999. And he did his MS in biotechnology also. And so here we have Dr. Chandan Kumar Sinha. We are all waiting for you for your talk, Dr. Chandan. So let's start off by the time your chair and co-chair will join. Thank you so much, Chandan. Thank you, Dr. Chandan.
Thank you, Manisha. I'll share my screen.
Yes, Vikram, any questions right now? Vikram has raised his hand. I don't know why.
All right, you can see my slide.
Dr. Chandan, we can start.
Okay, you can see my slide. You can hear me. Can you hear me?
Yes.
Sorry, I wasn't sure you could hear me.
Hi, Chandan. Now I'm able to hear you.
All right. So thank you, Manisha. Thank you for this kind invitation. And it's a great pleasure to connect professionally and to share some of our work that we've been doing for the past several years. I imagine it's a mixed audience with some teachers and a lot of students. So let's see what we do with this. The title of the talk is Precision Oncology, and I'll I wasn't very sure of the mixed background of our audience. So I've included this little timeline, which is to orient us to the world in which we live right now, which is the world of sequences of the human genome. Sequencing of the human genome was completed in early 2000. So 2001, some key publications came out where it was claimed that we have sequenced the human genome. It was hailed as a great success story of collective science where public and government and private participation declared almost like the excitement of moon landing, that we have sequenced the entire human genome, and people were wondering what exactly that means. So it's an exercise that was launched in 1990, so about 10 years time. International consortium of scientists from all over the world had come together with one purpose: to map every single base in the human genome, which is a collective of 3 billion nucleotides, to be sequenced, which means we know exact sequence of the entire human genome. And that became the starting point of a lot of research. And what I'm going to describe to you emanates very directly from having access to this reference human genome, which is our reference for a normal human genome. Among the many developments that followed in the wake of the completion of the human genome were actually technologies of more sequencing. In this cartoon, I'm kind of trying to demonstrate what exactly was achieved following human genome sequencing. So now, having access to a reference human genome, what some very enterprising and innovative scientists came up with, that we don't have to now, in order to sequence individual genomes, we don't have to do a 10-year exercise. We can do it faster because we have something to compare the sequence against. So a so-called human genome as a benchmark. Now we can sequence Manisha's genome or Sunil Gavaskar's genome or whoever's genome, because we need to sequence small stretches of DNA, map it to the available reference genome, and simply prepare a contig of an individual's genome as compared to the reference genome. And the idea is, wherever there are areas of mismatches, we can assess if those could be associated with some pathology, some germline aberration, or whatever. So that is the principle with which the so-called next generation sequencing kind of revolution came about in the following 10 years after the completion of the human genome. So from about 2001 to 2010 has been this immense period of growth in technologies and infrastructure, and sequencing costs plummeted. And I'm not showing you a whole lot of details on that. That used to be very fashionable just some time back, where literally for pennies, we started getting the amount of sequencing that was costing thousands of dollars just not that long ago. And it has happened in our visible research time after I've completed my PhD. All these developments have happened. So then, how it impacted our life and particularly cancer research. So in this cartoon, what I'm trying to convey is that initial machines to your left with a single solid circle is an indicator of how the human genome was sequenced, piece by piece, 1 unit at a time. That was followed by some technologies which could sequence large chunks of DNA very fast. And those machines are still operational, and they achieve what is called a depth of coverage. So a single position gets sequenced multiple times. And therefore, we achieve, instead of a position being read just once, a position gets read multiple times. And the hatch circles to your right bottom, they show you numerous very tiny dashes, which is to say, we are able to read a position. Right now, we are able to read a position hundreds of times over. That is the depth of coverage that we achieve, and it has implications for us, as I'll demonstrate to you in the next slide. So, one of the applications of this high throughput sequencing was we started sequencing cancer genomes. Cancer, as we know, is a condition of large-scale genomic aberrations. So we expect tumor DNA to have lots of mutations, copy number changes, and so many probably unimaginable aberrations affecting the DNA, as we know, have from some old times karyotyping. We know that there is polyploidy, there is aneuploidy, there are chromosomal rearrangements and whatnot. So now we have the capacity to go in depth and ask at the sequence level what has changed. So this cartoon is actually packing quite a lot, and I'll walk you through it because the rest of the talk will be simply a little higher level and relatively little more descriptive. So this is the learning slide. There are three things being shown here. We have on the left blue kind of diagram showing a patient's germline DNA. We are able to sequence it to some shallow depth because our germline is not undergoing too much changes. And we are concerned with cancer. So in cancer patients, germline DNA, we sequence it to some shallow depth that we know one position has been sequenced. So we refer to it as 5 to 15x means we have sequenced a position 5 times over or 15 times over, something like that. Actually, that is in real practice, it has come to currently I'm doing it even this position to a couple hundred times, if not more. So what that does is, if there are any germline aberrations in a cancer patient's genomic DNA, we are able to detect them and we are able to relate it to potential role in its cancer. And then we sequence the tumor genome to a higher depth because, as we know, tumor genome undergoes lots of chromosomal changes. So if a position is mutated in a percentage of cancer cells, not all cancer cells, and a different position is mutated in another percent of cells. Plus, my cancer cell tissue itself is not all cancer tissue. Part of it is normal tissue, part of it is vascular cells, part of it is stroma, and so forth. So I need depth of coverage, and I get it by sequencing to 70 to 100x I have shown in this cartoon. Currently, this number will be 500 times, 500x or more. And then I also sequence the RNA, where things get really interesting. So not only a genomic aberration is being mapped, we are also able to map changes in expression by sequencing the RNA. And I will show you some of its applications in the following slides. So essentially now what we have, germline DNA sequenced, tumor DNA sequenced, and tumor RNA sequenced. What all readout it gives us? We get readouts of structural rearrangements, copy number aberrations, point mutations, insertions, deletions, gene expression changes. And many of the readouts feed into multiple of these dimensions. And the end result is we get a fairly granulated, high resolution molecular landscape of individual cancers. A lot of these aberrations give us information on potentially what are the driving events that cause this cancer to happen, as well as they inform us on what targeted molecular therapies could potentially work in to reduce the tumor burden of these individual patients. That essentially is our stock in trade for so-called precision oncology. This strategy was launched in officially in 2011. So this year we mark 10 years of doing this exercise. As an operation, I'll walk you through its component parts. A patient comes to the hospital. We typically deal with patients with advanced or recurrent or metastatic cancers. They would be first offered genetic counseling and informed about the process of molecular analysis, as well as told that if we identify some germline aberrations, it will have a bearing not only for the patient, but for the family of the patient. So would they like to know such information? Or if they're not, then we would keep that information from them. Because some of the information can become anxiety inducing, lifestyle changing and so forth. So there are all kinds of considerations people have. So following genetic counseling, the tumor is biopsied or in some cases resected, and tumor tissue is banked, characterized, pathology evaluation is performed, and then sequencing is done. Following sequencing, we analyze the data. So we sit down with the bioinformatic pipelines output and try and interpret the data in terms of biological knowledge we have about these aberrations. And then report the results to the physician, who eventually communicates it to the patient. And some interesting cases, some novel observations, some confounding observations even, when those come, I present those cases to the Precision Medicine Tumor Board, which comprises of people doing the analysis, people performing the sequencing, bioinformaticians, clinicians, geneticists, bioethicists, and so forth. So it's a very full-fledged group of people, all stakeholders in patient care, and we collectively go over the data and come up with potential follow-up action items. So that is broadly the precision oncology workflow. And in terms of timeline, this whole process takes about a month. So when in the day when we initially plotted out this timeline, it was exactly 30 days. But nowadays we have become faster. So we can report out a patient's full analysis within 3 weeks. And in some cases, when there is very much urgency, we are able to further scrunch that time to shorter, about 2 weeks or so. I have already described the analysis that is shown on the top right in the slide. And then the tumor board, I already told you what all components are there. I'll walk you through a couple case studies to give you a real-time flavor of what we are doing here. And then I'll probably hit some broad stroke summary points. This one case came to us, it's been a year or maybe more, 35-year-old lady. She diagnosed with an extraskeletal myxoid chondrosarcoma. It's kind of a bone sarcoma. And when she diagnosed with this cancer, she also was found to be pregnant. So in the setting of pregnancy, she had this tumor growing right in her pubis, which was quite confounding in terms of how we care for this patient. Given the advanced stage of pregnancy, it was decided that we will wait out for the patient to first complete the pregnancy and then begin the intervention for the cancer. Luckily, the lady went on to full term. The healthy baby was born. And then meanwhile, a biopsy of a tumor was taken from her inguinal lymph node, which we analyzed. Some flavor of the quality control metrics, which are of interest if you are in the sequencing community. And suffice to say that mostly greens mean we had good data. Yellows meant data merited a second look, but it's all right. So we went on with the analysis. In the analysis, we break it into different components, as I already alluded to. We found a germline variant, which is considered a risk factor for certain cancers. So this will have a bearing on the genetic counseling for the family, which was followed up. It's not that terrible a germline finding. It increases the risk of breast cancer or certain other cancers by 1.5 to 3-fold. It's not like as serious as, say, for example, BRCA germline mutation and several others. So that was reported. In terms of somatic mutations, we found nothing much informative. Copy number analysis. So the blacks indicate there isn't much of a departure from the normal in this patient in terms of overall copy number, except some blue on chromosome 3 indicated there suggests a little copy loss, which we don't know much significance of that. And there is a gain of copy number, chromosome 11. That also remains somewhat nondescript. But here we found something that was very novel and very surprising and very exciting. We found this patient's tumor was expressing a gene fusion. This gene fusion actually involves NR4A3, that's an acronym for a glucocorticoid receptor. That is a known driver for these chondrosarcomas. But typically, this driver gene fusion is flanked by some other upstream partner. Typically, that will be an Ewing sarcoma receptor gene, EWSR1, or it will be a TFG gene, or some others that are shown in the cartoon in the box below. Never before a NR4A3 fusion had been seen that was driven by PGR. And some of you in the field would know PGR stands for progesterone receptor. And you can imagine this lady was pregnant. So she was being quite progesterone rich at the time. And here is a progesterone receptor gene driving the driver of her tumor. No wonder it was such a bad acting, aggressive cancer that was growing along with the baby. But now that we know that progesterone receptor is driving this tumor, we knew what could possibly work for her, because progesterone receptor is actually driven by estrogen receptor. That's biochemistry we have known for probably 100 years, or maybe not that long, but close to that. So estrogen drives progesterone, and estrogen receptor inhibitors are used for treating breast cancer patients for a number of years. And there are several safe and effective estrogen receptor inhibitors, including tamoxifen, which is used quite relatively routinely. So we speculated based on our observation that in this patient's tumor, if we treat with tamoxifen, which is a very standard of care therapeutic, the tumor might regress. Further supporting this hunch was the fact that we noted the expression level of estrogen receptor, progesterone receptor, and the glucocorticoid receptor. All of them were very high in the tumor, as seen by these red marks on a histogram plot of the expression of these receptors across our compendium of more than 3,000 cases. Virtually in this tumor, these receptors are being expressed at the highest levels that we have seen in our whole compendium. So that was a very, very strong indicator that tamoxifen probably should work. And mind you, if we do not have this option, if our chondrosarcoma was being driven by any of these other common drivers, we have no therapies that are known to work against any of those. So a typical chondrosarcoma, there are no known effective therapies. There are no known FDA-approved therapies. And here we are able to treat with tamoxifen. And the story is actually a happy story after all. And this patient has been treated after our finding for past 3 years. And despite intraoperative rupture and previously aggressive recurrences, there is currently no evidence of disease. So these happy instances are not the routine. They are somewhat of outlier cases, but it's good to see the sequencing-driven cancer therapy being actually effective when stars align. And one example I'll share with you, it is a very intriguing example as well as in a different dimension. So here, not only we informed therapy, but we also informed diagnosis. This patient came to us, 65-year-old male with a poorly differentiated adenocarcinoma of unknown primary, which means we see a cancer, we see it in the rectal area of this patient, and histologically, we can't nail it as to where this tumor actually belongs. What is the primary tissue of origin? We simply see it in the lymph node in the perianal area, and histology is very non-definitive. Another thing that we learn about this patient is that there is a family history of cancer, Ashkenazi Jewish ancestry, and the patient's sister had premenopausal breast cancer, which means relatively younger age breast cancer. Our entire workflow is operational now, and we find in germline variations one mutation in BRCA1. That is a very strong player. It's a famous gene, we all know. It is associated with very high chance of getting breast cancer. But we are dealing with a 65-year-old male, and the tumor is in the anal area. So let us see what else we see. So in copy number changes, we see an amplification of ERB2, which is also known as HER2, and that also shows a very high level of expression. So here we have a clear-cut case, and HER2 is HER2 positive tumors are expected to potentially respond to Herceptin, which is an antibody, monoclonal antibody effective against, and particularly effective in breast cancers against tumors which are amplified for this HER2 gene and overexpress HER2 as well. So this was a very useful finding. But the most surprising thing of all, we have an algorithm to type the tumor's tissue of origin. We call it cancer of unknown primary. We have a certain methodology to it and certain logic of making it. We don't have time to get in the details, but suffice to say that our cancer of unknown primary identification algorithm called this particular tumor as breast cancer. It had zero doubt that this is breast cancer. Now, we have already seen in the clinical workup that this patient's MRI, abdomen, pelvis, CT chest, colonoscopy, everything was clean. There was no trace of what could potentially be wrong. And here, our algorithm says this is breast cancer. But data is data, and we had to go with it. So we further dug into it, and sure enough, we learned that there are mammary cells that reside in the anogenital region of both males and females. These are some residual cells which during development, identical to mammary cells, they live in the anal region, and some of them live on for life, and they can become cancerous. And when they become cancerous, they are essentially breast cancers. This patient is a case of male breast cancer growing in anogenital region. It is the rarest of rare cases, but we were able to detect it through our sequencing pipeline. Not only identify its diagnosis, identify the germline driver, identify a therapeutic target. And this patient also went on to receive Herceptin and has responded beautifully. So that's the power of the power and the satisfaction and the impact of this precision oncology on individual patients. And this is a kind of a screenshot of a report that we put out for every patient. And I think that number by now is probably more than 3,000 overall in the past 3 years, in the past 10 years, I'm sorry. Now, the big picture, as I promised you, this is a paper we published where we reported 500 cases of this precision oncology sequencing and a summary of all the different tumor types, different metastatic sites, and what big picture observations that we gleaned. So first of all of our patients that we register for this study tend to be advanced cancers, metastatic cancers, and not surprisingly that they have high burden of metastasis. What we found a little more surprising was that more than 12% of those cases had some pathogenic germline underlying condition, much more than patients coming to us with a family history of cancer. When there is a family history, one suspects that there may be a germline driver. But without a known family history, cancers are being driven by germline drivers. And that told us quite strongly that germline aberrations need to be screened for in larger number of patients, probably more so when there is metastasis, because that study, that knowledge has a bearing on the entire family. This paper we published relatively recently, earlier this year, I think. And here we reported our clinical care of first 1,000 patients that have gone through our pipeline. And we narrated the different varieties of clinical actionable diagnostic findings in our studies, and how many patients went on to receive therapy, how many benefited, and how many diagnoses were refined or corrected or identified. So it's a very good paper, and you can look it up in PubMed or elsewhere. And if you are interested, I can provide the links to Manisha. In this paper, we reported the sequencing of about 100 pediatric cancers, again finding a more than expected burden of germline variant aberrations, and numerous cases where the children went on to receive precision driven targeted therapies, and they went on to, many of them went on to respond what we call exceptionally. So they were exceptional responders, and many of them were routine responders. And in this process, we have identified numerous novel observations. Some of them we report when we get a bulk of them. So in this, for example, in this study, we reported a number of FGFR fusions. It's a receptor kinase, which are very difficult to speculate which cancer type. So across a large number of different cancers, we found a large number of different varieties of FGFR fusions, many of which respond to FGFR inhibitors, and some of them have since then gone on to achieve FDA approval. So it is another one of our good success stories. In this slide, I have tried to encapsulate another angle that we are exploring for our cohort, is that a large number of our cancers, actually 65% in the slide I'm showing you, are by definition rare cancers, which according to American Cancer Society are those cancers which are seen in 6 or fewer cases per 1 lakh people per year in this country, in United States. So these are really small numbers, but a large number of such patients get these rare all different kinds of rare cancers. So the total number of patients who suffer from some or the other rare cancer, that number is actually not very rare. It's huge. But individually, there isn't that much information about them because there aren't many clinical trials conducted, or there aren't any statistically robust cohort data because they're rare. But so then we are thinking that we have a big compilation of them and a large number of those rare cancers, we have fairly good numbers of them. So we are able to actually come up with some good, rigorous, statistically significant bioinformatic analysis and provide some new insights. So this I'm sharing with you just so we will be it's an active area of investigation, and hopefully we'll be coming up with some good publications on this shortly. In this slide, I'm kind of trying to convey that it's important to sequence the cancers across all the different dimensions that I have described, because as I've marked in this matrix, different patients, actionable informative observations come in different layers of data. They all don't come as mutations. They all don't come as germline variants. And we don't know in which dimension, which tumor will provide us actionable informative knowledge. So that's why we emphasize the integrative aspect of this precision oncology. And I'll end with this last case study to make a point that is another area of growth for us. It's something that we have not been doing, but this is a case to make an argument that this needs to be done. In this case, we have a renal cell carcinoma, which had an unusual pathology, which is like apart from clear cell renal cell cancer cells that you can see easily in the slide for those who are pathology inclined. It also has some sarcomatoid rhabdoid component, which makes it interesting to the pathologist as well as to us molecular analysts. This patient was known to have a germline variant in a gene called BAP1. It is a BRCA-associated protein 1, but the variant is not of a known significance. It's known in the community as variant of unknown significance, or VUS. However, this patient's father had died of metastatic RCC, the same disease as the child has. That raised a flag because this BAP1 mutation that we noted in our germline variant analysis is actually associated with as a risk factor for renal cancer. What are the odds that the father has a BAP1 mutation, the son has a BAP1 mutation, father died of metastatic renal cancer, the son has renal cancer? What are the odds that this variant of unknown significance is actually a pathogenic variant? That was the question. And we have no way of knowing it, simply seeing the presence of the variant. Now, luckily for us, one of the dimensions in which we are not performing our analysis is proteomic analysis. We are not testing the protein. I have only described to you germline, mutations, copy number changes, RNA, but not protein. However, in this case, a BAP1 mutation, if it is pathogenic, has a signature at protein level. By immunohistochemistry, we can see that if the BAP1 is mutated in a way that it's deleterious, the protein expression is lost. That can be detected by immunohistochemistry. So we went ahead and triggered immunohistochemistry analysis, and with my pathology colleagues, I found it was very satisfying to see. Indeed, this patient's BAP1 missense mutation was pathogenic because, as you can see, it's a busy slide, but upstairs you see H&E staining on the top left. On the right is a traditional marker of renal cell carcinoma. It's all brown, which proves that it is renal cell cancer. On the bottom left is BAP1. It is mostly empty. Blue is just the baseline stain, and there is no brown staining. Whatever little few brown cells are seen, those are background cells. And this thing is not being shared because I use this slide for some other more academic presentations as well. And this protein, mysterious protein X, is our discovery that has been obtained from some additional bioinformatic analysis and were nominated by large-scale proteomic analysis. And we have discovered that when BAP1 is lost, this protein is gained. And the cool thing about this new protein which is gained is that there is an inhibitor available for this protein. So it's hard to target something which is lost. But if something is gained, we can inhibit that and hope to treat the cancer with this. So this new information came only by looking at the protein. And we are using this particular discovery to make a case that we need to do a little more systematic and routine analysis of proteins apart from the genomic analysis in our precision oncology efforts. And this is probably, I've forgotten, I've included this as well. It's a very good example of why proteomic analysis can be super informative. This is a case of lung cancers, and this we published recently in Cell. Here, ALK fusion is a known driver of a subset of lung adenocarcinoma. And typically we detect this fusion by analysis of either DNA or RNA. Here we went on to look at the proteins. And we found that whenever there is gene fusion expressed in our cases, we found high expression of protein as well as a very specific phosphoprotein expressed only when there was fusion. This phosphoprotein in a fusion-specific way was validated by immunohistochemistry in multiple fusion-positive samples. And what made it really exciting is that when we analyzed a larger number of samples, we in fact found many cases where the fusion is present, but the phosphoprotein is not seen. Or in one case where fusion is not there, but this phosphoprotein is seen. This leads us to a hypothesis that not all fusion positive cases will respond to inhibitor. And in cases where the phosphoprotein is seen independent of the fusion, that tumor probably will respond to ALK inhibitor. The former situation is known to clinicians. They go into a diagnostic laboratory, fusion is seen, but not all of them respond. We believe we now know why. As well as we believe that in some cases where fusion may be negative, they may still be candidates for benefiting from this inhibitor. So that's the power of studying the protein. And we are making a strong pitch to find a way to do this in a routine fashion. And this is kind of our wish list for essentially grant applications and so forth, that we want to analyze proteins at a large scale. So I think I want to stop here. And a large number of people, I work in this group, which is led by Arul Chinayan, and I'm part of the genomics group. And I have named several of the other folks here who are organic and instrumental in this entire operation. And happy to take any questions.
Hello. So it's my privilege to read out the questions which are placed on the chat box. So some of them I will be actually reading out. And Dr. Purohit. Yeah, so Mr. Rajkumar is asking whether it is possible to assess the vulnerability of upcoming generations to develop cancer by screening the genome of sperms and ovum.
Wow, that is that to my knowledge is not routine right now, but I would say theoretically it should be possible. But another thing is, uh, if I got the question right, it is asking the possibility of, and what we do say in our field is the risk of based on knowledge. So, for example, one of the mutations I shared in my presentation in CHEK2, a pathogenic mutation in that gene statistically increases the chance of a person getting breast cancer by 1.5 to 3-fold, which is to say, in a population, for example, I know the statistics for the USA. There is 1 in 8 women is supposed to have a risk of getting breast cancer by a certain age. That risk, if it is 1 in 8, it becomes 2-fold higher, which means the risk will become 1 in 4. So it is a statistical thing. On the other hand, if there is BRCA mutation, and there are nomograms available for various populations, if there is a BRCA mutation, there is a much higher chance of getting a breast cancer, and that chance increases with age. Unfortunately, such data are not uniformly available. So in some countries, particularly Caucasian countries, these data are a little more evolved, but I don't know if we have similar data from, say, Indian populations. And a lot of cancer chance depends on environmental factors, and it's a little more complicated.
बेटा अभी ना मैं अभी बिजी हूँ आपका जो भी है आप प्लीज टेक्स्ट कर दीजिए ठीक
Yeah.
है
So may I take the second question? This is by Arushi. So she's asking, she's inquiring actually, tumor has zero effect on pregnancy of a lady, if so, or any special kind of treatment has been given. So this is her question.
For that particular case, no, the tumor there was nothing that could be done to the tumor because that might affect the baby. So other than biopsy, it was just let be. It was monitored, and the mother's health was monitored, and the baby was presumably unaffected. And it went on to deliver the baby. So to my knowledge, no, and nothing was done till the baby was born.
Okay. Kiran is asking, what are the possible role of transposomes in developing cancer?
That is an area of investigation. I'm not sure that something very definitively is known, but you should look up in publications and you will find something. One thing I do know is that there are some endogenous retroviral elements, human endogenous retroviral elements, they are called HERVs. Those have been noted to drive certain gene fusions. So, in the chromosomal rearrangements, if some HERVs come to drive a potential oncogene, then that's a genomic accident that caused a cancer to be driven. And I know for a fact some gene fusions in prostate cancer that are driven by HERVs.
Yeah.
Okay.
Yeah. So, Indranil is asking, is there any role of bacteria in cancer detection?
Role of bacteria in cancer detection.
Yeah.
Well, first of all, you would know that bacteria actually causes one cancer. it doesn't cause too many, known to particularly cause too many cancers. Viruses, many viruses are known to cause cancers, but Helicobacter pylori has been officially associated with causing gastric cancer. Actually, the discoverer of this connection from Australia went on to receive the Nobel Prize in Medicine some years ago. And now, having said that, increasingly people are associating the microbiome with various aspects of cancer, including its effect on treatment response. And some very exciting recent research has come up. I think I am able to share two vignettes with you. One of them has come very, very recently in the last month or so in prostate cancer. Now, prostate cancer is one of the more treatable cancers for good part. So, and the easy remedy is, and more than one Nobel Prizes in that area have gone. One of them actually from University of Michigan, is that the simple treatment of prostate cancer is castration. The original castration was physical castration, but later chemical castrations, which means essentially drugs that will neutralize the testosterone in the patient. Prostate cancer feeds off testosterone, no testosterone, and the cancer begins to die. So that is the standard of care for all prostate cancers, and it's fairly effective. But in many cases, that effectiveness wears off and the cancer becomes so-called castrate-resistant prostate cancer. Chemical castration has been done. All the various generations of chemical castrations are done, but the cancer evolves and it has become castrate resistant. So very recently, this paper has come, which has shown that possibly bacteria are involved in causing castration resistance. In that when the patient's system is challenged for lack of testosterone, somehow the system makes the gut microbiome to start synthesizing testosterone. So that connection is very, very recent, but it came in a very high profile journal. So I'm sure it will be followed up. So that is one thing. And second thing I promised you is that in pancreatic cancer, which is another very lethal cancer, some report, and again in some very high profile journal, they reported that the standard of care therapeutic for pancreatic cancer is a cell cycle inhibitor called gemcitabine. Now, this gemcitabine was shown to be metabolized by pancreatic microbiome to an inactive form. And that was proposed as the reason why in a large number of patients, they do not respond to gemcitabine and succumb to it. So the microbiome does have a strong role to play in cancers in many ways, some of which are not yet fully characterized. But I think the original question was, does bacteria cause cancer? And well, I gave a rather long answer, but the short answer was, in one case, we know for sure, otherwise, probably not.
Yeah. Ranjana is asking if these kinds of, how far this is being used in India. Probably she is trying to talk about the new developments of drugs which are being used abroad. Is there any practice going on in India? Probably this is the question which I assume. Yeah.
I'm going to guess that is not the question. I'm going to guess the question is about this concept of precision oncology, this molecular characterization and matching the genetic aberration to a targeted therapy. If that is the question, I think to my knowledge, it is available. There is a lot of sequencing happening in India. I know that. But how far a comprehensive program like this is operational, I'm less sure about. I do know in South, in Bangalore, in Hyderabad, in Chennai, I have, I think, more scientific connections there. So there is some activity going on. But again, it comes in at different scales. And to my understanding, the scales in India are probably not as well evolved as they are out here, and probably in other little more richer countries like Europe and Australia.
Amrina is asking, I just wanted to ask that do you think that there is a significant role of polluted environment in triggering the incidences of cancer in humans and animal population?
The general consensus is that that is very much the case. It is a bit hard to establish causal relationships. You know, and there's fascinating story about this in a very, very engaging, informative, exciting book, actually, that you probably should be able to find it online for free. It is considered a classic. It's by an Indian author, Siddhartha Mukherjee. And the book is called Emperor of All Maladies. And an entire chapter or so is devoted to this concept that it took about 100 years of intensive research and advocacy from a lot of people to make a case that cigarette smoking causes cancer. We take that knowledge for granted nowadays, but it hasn't been easy at all. And there were some very strong forces pitted against this idea because it was connected with business and money and whatnot. And you would imagine that cigarette is relatively very intense treatment of pollutants at individual level and for long periods of time. So effect of everyday exposure to pollution, how it is causing cancer, that becomes a very challenging research subject. But the general consensus is yes, of course. And one example is in the US, particularly, mesothelioma. It's a kind of a very bad lung cancer. Is caused by asbestos, which is used in building material. At least it used to be used for insulation. And asbestos got connected with mesothelioma by similar research strategies that were that connected smoking with cancer. Another fascinating story about this is actually the discovery of cancer, environmental pollutant and cancer. It goes actually to Britain. And those days in the colder parts like Britain, there would be fireplaces and there would be chimneys to convey the smoke. And there would be little children, they're by definition tiny children, who would be used as chimney sweeps. They literally have to crawl through those chimneys and clean up the soot that accumulates in those chimneys. And that used to expose them to this extremely toxic carbon repeatedly, and they were coming up with cancers. So environmental pollutants causing cancer, this connection is long known, but individual associations, they become tricky if the intensity is not super strong. And of course, you know the story of radiation causing thyroid cancer. Chernobyl was a hotbed of that. And earlier, Japan, the Hiroshima nuclear bomb were followed up by plenty of cancer cases. So, yeah.
Yeah, I think there is no more questions. So may I now ask my colleague to introduce you, because of paucity of time and there was a technical glitch. That's why we could not
No
introduce
worries.
you in the beginning. So may I ask my colleague to introduce you first?
Thank you, Dr. Jageshwar. It's my honor and privilege to say a few vote of thanks to Dr. Chandan, as well as to introduce him. So first of all, let me thank you for such an informative and interesting talk. Everybody knows that cancer is a cause of concern for everyone. And it was very well depicted from your presentation that you have a huge experience. You have huge experience in high throughput genomic and transcriptomic analysis based on next generation sequencing. The primary area of research is functional genomic analysis of cancer towards discovery of disease biomarkers and therapeutic targets. In a career spanning over 20 years, Dr. Chandan has been involved in the characterization of biomarkers of prostate cancer, breast cancer, systematic discovery of gene fusions by RNA sequencing, discovery and characterization of recurrent gene fusions, and signatures of cancer-specific transcripts. Using transcriptome sequencing data to define precision therapeutic agents, he has been studying, characterizing kinases that display outlier expression in individual cancer samples as potential targets. So basically, he is an alumni from Delhi University. He was in Department of Zoology. Then he did his master's from biotechnology. He did his PhD from Indian Institute of Science, and his postdoctoral research is from University of Michigan. His career started as a senior research scientist in the Institute of Bioinformatics at Bangalore. Then in 2004, he was appointed as a scientific officer at Advanced Center for Treatment Research Education in Cancer, that is ACT-REC. In 2006, he worked as an assistant professor at the same center. In 2007, he worked as a research assistant professor in the pathology department, Michigan Center for Translational Pathology, University of Michigan. And since 2019 till date, he's working as an associate research scientist, pathology department, Michigan Center for Translational Pathology, University of Michigan. He's an active member of he's an associate member at American Association for Cancer Research. He's a life member of Indian Association for Cancer Research. He has been awarded with several honors. To name some, in 2009, he was awarded with Pancreatic Cancer Research Award. Then in 2005, he was awarded with Young Scientist Award by Department of Science and Technology. In 2011, he was awarded with American Pancreatic Association Young Investigator Award. In 2013, he has been awarded with University of Michigan Medical School All India Institute of Medical Science Collaboration Travel Grant, and there are many more. He has contributed a lot in cancer research and high throughput genomic analysis, and he has many research articles in very, very reputed journals like Nature and Cell. This is to name some, but he has several such reputed articles. Many of his research projects have been funded by NIH. So it was really very encouraging to know that you are an alumni of Delhi University, and each of our students who are really attending this conference, they are going to be really inspired by you. And it was really very nice of you that even during an odd hour, you have accepted our request and you could spare some time to share your knowledge and your research with us. With these words, I will not take much of the time because probably we are short in time. So I would like to thank you from the core of our heart, from the entire organizing team. We would like to say thank you. Thank you very much.
Thank you.
So here's a token of appreciation for you, Dr. Chandan. It was a beautiful talk, a very lucid one, and everybody is very happy to hear you. and they have many queries and they would be maybe disturbing you in the near future. So please attend to their mails.
Oh, sure.
The request.
Yeah, happy to.
Thank you. It was such a wonderful session. Wonderful. Thank you. So proud to
Thank-
be associated with you, Chandan.
Hey.
So proud. I'm so proud.
Thank you. Proud of you.
I didn't know at all.
Okay.
Chalo, thank,
Take
please
care.
go off the seat now. Thank you so much.
Thank-
God
bye.
bless you.
Thank you.
God bless you. Thank you. Bye. Thank you. Thank you to the chair and the co-chair too. Thank you. Thank you, Archana. Thank you, Dr. Purohit.
Thank you, ma'am. Thank you. Bye.
Bye-bye.
Thank you, ma'am, so much.
Thank you so much. So now we have Rahul and Sushma, and the next, I think it's time for the next speaker. Sir has already joined. Good morning, sir. Good morning, sir. Sir, how are you?
Good morning. Everything is fine. Yeah.
Nice to have you here with us again, sir.
Thank you.
We're always eager to listen to you.
Thanks.
Thank you, sir. Thank you. So we have the chair and the co-chair for you, sir. Just a minute. Ma'am has joined. Ma'am Rita Rath, ma'am is a very senior. We have people to introduce ma'am and Monica also. Ma'am is in Dayal Singh College. She's a very senior person, and she's a researcher herself. And Dr. Monica Sharma from Miranda House. They both are doing research, sir. So, Dr. Kiran, are you introducing them, ma'am? Introducing them, Kiran?
Good morning, ma'am. I'm introducing our chair and co-chair. So good morning, everyone. I welcome you all in this session of the International Conference. Today, as chair, we have with us Dr. Rita Rath. Ma'am is an associate professor in the Department of Zoology, Dayal Singh College, University of Delhi. Her expertise is in the field of entomology, animal diversity, and animal behavior. Ma'am has an experience of more than 30 years. Along with ma'am, we have Dr. Monica Sharma as co-chair. She is an assistant professor in the Department of Zoology, Miranda House College, University of Delhi. Her expertise is in the field of immunology and physiology. I welcome you both in this session. Over to you, Dr. Rita.
Thank you so much, Sushma. Thank you for giving us this opportunity. So on behalf of the principal of Deshbandhu College, Professor Rajiv Agarwal, and the entire organizing team of the Virtual International Conference on Health 2021 on Cancer Biology, its advances and challenges, I extend a very warm welcome to you, Professor Gajendra Raghav, to this session 7 of this 3-day international conference. A very warm welcome to you, sir. A very warm welcome to all the participants as well to this conference. This conference is organized by Deshbandhu College, University of Delhi, in collaboration with Indian Council of Medical Research, Government of India, and the National Institute of Pharmaceutical Education and Research, NIPER, Hyderabad. Today, Professor Raghav will be delivering a talk on computational resources in cancer. He is an eminent renowned professor working in the Department of Computational Biology at the Indraprastha Institute of Information Technology, Delhi. He received his MTech from Indian Institute of Technology, Delhi in 1986 and PhD from Institute of Microbial Technology, IMTech, Chandigarh in 96. He worked as a postdoctoral fellow at the University of Oxford from 1996 to 1998 and as bioinformatics specialist at University of Arkansas for Medical Sciences, UAMS, USA in 2003 and 2006, and has been a visiting professor at POSTECH, South Korea in 2004. He has worked for around 30 years from 1986 to 2017 at different level scientific positions in the field of bioinformatics at the CSIR Institute of Microbial Technology, Chandigarh. Professor Raghav's group has published more than 300 research papers in reputed national and international journals and developed more than 300 in silico products such as web servers, databases, and software packages. This is indeed an incredible achievement and calls for an applause, being the highest contribution by any single group in the world. So it is indeed a privilege for all of us to hear him today. He is a strong supporter of open resource software, web servers, and therefore all services developed by his group are free for academic use. These web-based services are heavily used worldwide, and it is incredible that they receive more than 150,000 hits per day. It is indeed very heartening, sir. He has been adorned by many awards, a few among them being the Shanti Swarup Bhatnagar Award, awarded to him by CSIR in 2008, the National Biosciences Award by the Department of Biotechnology in 2006, the J.C. Bose National Fellowship from 2010 to 20 by the Department of Science and Technology. He is also a fellow of many major scientific academies, such as Fellow of National Science of Academy, FA Science, and FNA, to name a few. So without further delay, I would request Professor Raghav to kindly deliver his talk. Looking forward, sir, to your enlightening, informative, and interesting talk. Over to you, Professor Raghav. Thank you.
Thank you very much for nice introduction. And this talk will be more like a advertisement rather than serious science. I will talk about the different resources developed by our group in the field of computers in cancer, actually. So the person who are working in the field of the cancer can use these tools and resources for their research. As already Madam told, that all resources are free from our group, actually. That's our motto, that research should be affordable. What happened in countries like India, it's very difficult to afford the research sometimes due to the grants, basically. So what we are doing actually over the years to promote the freeware so that everything is available free to the community. So I'm sharing my slides. Okay, so the talk is on the computer resources for cancer. So I will just cover few slides for introduction. You all know about the cancer. So major steps in cancer is just first part is how to detect it. We call the screening or diagnostics. There are a number of technology are already there in the market. You can use one of them. But what we are going to talk about the genomics based technology. Then the next question is, once you know the person is suffering from cancer, you want to know whether person is on the high risk or on the low risk, because treatment is different when the person is on the high risk. So you have to predict the risk, actually. That's a different type of biomarkers, you need it. And then ultimately you have to provide the treatment. So I will cover these different topics one by one. You already know the cancer hallmark, so I will not introduce this topic, property of the cancer, because most of the audience, they know about it very well. This is important for the persons who are in the field of the biological sciences to understand the different terms, actually. You know that genome is in the nucleus, and when we sequence the whole genome or whole DNA of a cell, then we call the genomics. In case of the human, this is a 3 × 10⁹ base pairs, actually. So that's the study is called genomics. Whole part of the genome is not translated into the proteins. There are certain regions which translate into the proteins, and that is called gene, protein coding genes. And they convert first to the mRNA and then to the protein. So mRNA in a cell is called transcriptomics. These terms are important to understand the things clearly. As well as DNA is bound around the histones basically. There is a modifications, different type of modifications are there, and this area is called epigenomics. When the transcriptomics, this mRNA converted into the proteins, so all proteins in a cell we call proteomics. We call proteome, and if we study it, then we call proteomics. Then there is a post-translation modification like glycomics and others. Then we call the glycomics or metabolics. So there are different terms in there. It's very important in today's time, if you are a student, to learn these terms so that you can understand the literature easily. The question is, what type of resources our group provide to the community in the field of cancers? Actually, we work in the different field, but I'm just focusing on the major tools, major resources we developed in the field of cancer. Okay, so if you have the interest, you can go to my website and you can get all the servers. There are more than 250 servers are there, or resources are there. You can use them. All are freely available. So first I will talk about the databases. Resources means databases. I will focus on that. So there two databases I will describe it, CancerDR and Herceptin. Then I will talk about the gene databases or biomarkers. Then I will talk about the drug delivery databases. Then cancer biomarkers databases. So I will talk about these resources first, and then next I will talk about the different type of prediction methods or different type of biomarker predictions. Now come on to the cancer drug, because everybody know that there is a drug resistance in cancer. What happens? You give the drug, it works for some time, and after some time it becomes resistant to the cancer. It no more effect actually. Then we have to change that. The question is, can we find out which drug will be suited to best to the person, to the patient, based on its genomic profile? Genomic profile may be its transcriptomics data, genomics data, or whatever the genetic information is concerned basically. So there are a lot of talk about this area, but no database was there at the time when we developed this database actually. And the idea was just to collect and compile the information so the user can get the complete information from a single source. Please remember one thing, creating database is not a big thing, but maintaining the information up to date is a major challenge actually. Okay, so this database maintains the information about the large number of cancer cell lines and the number of drugs tested on these cancer cell lines. So IC50 values are there actually. And we maintain the complete information, their genomic profile, let's say gene expression, mutations, variations, copy number variations. Okay, so different type of information we maintain in this database. And this data is not generated by us. Please try to understand, this is generated by the biologists. We only pick up from the literature, we compile the information, so you get the complete information from a single source. Rather than going here, there, and other places, you will get the complete information from a single source. So we allow to predict the structures of the target as well as alignments of the targets. So different type of facilities are there. You may have a look by visiting them. There is another thing actually, in breast cancer, one of the popular treatment is by monoclonal antibody, and that's name is Herceptin basically. Herceptin monoclonal antibody is heavily used to treat the breast cancer patient. But it has been shown that for some patient, it works, some patient it doesn't work. And it's very costly affair. If it fails, it's a big economic loss as well as loss of the time actually. So what we did actually, we collect all the information, including the genomic profile, where it has been shown by the biologist that when it failed, then this was the genomic profile. When it worked, this was the genomic profile. So you can find out the relation. So if you have the genomic profile of the patient, you can simply check whether it will work or not. So without giving to the patient, check in advance whether it will work or not. So this database is available. Now these are the gene-based databases. So here it's for the methylation database, where we have work on the pancreatic cancer, and methylated genes have been stored here, maintained here. Please remember one thing. All web servers are so user-friendly, even if you don't read it, you can easily access the information by searching, browsing, summary. So complete international standard has been found, and all databases, whatever I am describing to you, are already published in highly reputed journals. So citation is not a problem. And most of the tools are heavily cited worldwide actually. So it's not like local tool sort of things. Whatever I am describing, it's well validated. It's all reviewed work actually. So when you are using your work, nobody will question it why you have used this server actually. Then this is a another database which maintain the epigenetic modifiers basically, and provide the complete information like mutation data, copy number, expression data, gene expression associated data. So complete information is there. Similarly, this is for the apoptotic proteins basically. As you know, the apoptotic proteins, the apoptotic pathway is very important in cancer cases particularly, because this is not functional properly. That's why there is no program direct actually. So it's important to understand that what is going in the cancer patient. So this database maintains the information about the different cancer patients or cancer cell lines in and their apoptotic pathway. What type of changes are going on? That's why it's not functioning as it is supposed to function. So this database provides all the information related to apoptotic proteins in context of the cancer. This is a new thing. Sometimes students come up with the new ideas actually. So this resource to explore the therapeutic potential of oncolytic viruses. Though we know that virus is dangerous to us, and we already know that number of viruses are responsible for the cancer. Okay, but there are other way around is also there. So here we are a resource to explore the therapeutic potential of oncolytic. Means what we are saying that there are viruses which can kill the cancer or which can be used to treat the cancer actually. What we have compiled here, all the resources related to the virus, where the virus can be used to manage the treatment of the cancer or to treat the cancer, has been compiled here. So you get that information. These are the unique databases in the world. Nobody have done earlier actually. This is important to understand that most of the time our Indian community, we are busy in using the others resources, but we have changed the trend. We are developing the resources so the world can use the resources developed by Indians basically. That's our motto basically. We want the Indians should be in the contributor also, not only users only. Because if you want to earn your respect in the life, you have to give to the public. That then the public will respect you. If you are always taking from the, let's say, resources from the others actually, nobody will give you respect. Anyway, so this oncovirus database, oncolytic database, contain these number of records, research articles. So a lot of information had been compiled actually. So now you can understand, if you start to read the papers and compile this information, it will take a long time. My students work day and night actually, and use a different type of softwares to compile and collect and compile the information and then put it here actually. So you can use in your research. This is another type of resource. That's a cancer liver basically. Cancer liver is, we know it's very dangerous actually. It's a very popular, it's harmful. So what we did actually, we collect the different type of cancer data collected from literature. Please remember one thing. We only maintain the data which is experimentally validated by researchers. We are not maintaining any predicted data. We only maintain in our databases that's basically experimentally validated by researchers. So when you are using this data, you are sure, and the corresponding reference is there, that this data has been already validated. Basically. So that's not a problem basically. And all work in these databases are manually curated. It's not a computer-oriented sort of thing. You just collect relevant word, whether it's there or not. We read each and every paper and we put this information to you. So you get the clean information and complete information. So this maintain, cancer liver maintain all the type of biomarkers that may be different type of biomarkers, maybe based on the protein, based on the RNA, sort of things. Then we have another database called cancer, and a database of cancer associated enhancers. In regulatory regions of the genes, there are enhancers which are responsible for the gene expression. So we collect that information from the different sources, different databases, and then we create the database of cancer enhancer database, and we also shown that performance, that survival association with the cancer enhancers. So it will help you to identify the enhancer-based biomarkers. So and database is available, you can use it. So far, I talked to you about some of the important databases developed by our group. Now I am talking some of the softwares, prediction methods developed from our group. So now question is, what is application? Database is important when you are interested in studying the particular type of information, rather than getting the information from literature, which is difficult sometimes, you can get from our resources. That's the advantage of the databases. In case of the prediction, it developed the models using machine learning technique, or what we call nowadays artificial intelligence based models, where they can predict the right targets basically. So these are some of the important tools. Let's say personalized vaccine and drugs. I hope you understand the concept of the drugs and vaccines. You know that vaccination is a process where you artificially train your immune system. So in future, immune system itself can fight against that particular disease. For example, nowadays you have the different vaccinations against the coronavirus. It means we are training our immune system in such a way that if next time we got the infection from the coronavirus, then we know how to defend it actually. And another concept is drug, where we search the different. So here, these two data servers allow you to predict different type of vaccine candidate against the cancer, as well as the drug candidate, prioritization of drugs basically. Then this is another server which help you to predict the promiscuous cancer drugs basically. And these are the biomarkers where you predict what is the stage of the cancer, what is the progression of the cancer, okay? And there are some other methods which provide some therapeutic peptide or therapeutic molecule against the cancer basically. These are the anti-therapeutic peptide protein. Okay. So I will go one by one. Cancer talk. We all know it's difficult to develop the vaccine against the cancer. Why it is so? Because it is easy to kill the enemy when enemy is from outside. Let's say if somebody, let's say any country, China, Pakistan, or any neighbor country attack on India, it's easy for India to revert back or to respond to the attack basically, or to counter back attack. Because we know it, they are outside of our country. But think about the terrorists who are inside our population, and some of the people who are misguided or whatever the reasons, they are clean. Then how you will handle them? Because it's very difficult to recognize the normal person and the terrorist basically. Same way, the cancer cell and normal cells, it's very difficult to discriminate them actually, because cancer cells are mostly generated from the normal cells. So they look like the same, the genetic feature is the same. How you will identify them actually? That's a challenge. That's why it's very difficult to design the vaccines. So what we propose here, take the cancer cell, sequence it, and compare it with the normal genome which is achieved from the normal cells, and find out the regions, find out the protein regions or antigenic regions which are in the cancer cell but not in the normal cells. And then these regions, if they can activate the B cell and T cell, then this can be serve as a vaccine candidate. So based on this principle, we have developed this CancerTop, and CancerTop have the simple concept that it take the genome, then it take the vaccine candidate, cancer antigens basically, then generate the 9 mers, because 9 residue peptide is sufficient to activate the B cell and T cells basically. And if present in the reference genome, means in the normal, reference genome means normal, then delete it, remove it. Otherwise, take it further, check whether it have the different type of epitope, like HLA binder, class 1 binder, class 2 binder, epitope. If yes, then this is can serve as a potential vaccine candidate. So this is a good example of personalized vaccine concept basically. So we have given the both partially personalized and fully personalized. Please remember one thing. In this talk, it's not practically possible for me to cover the depth science. If I start to cover the depth science, even single paper, single server will take 1 hour actually. So that's not practically possible. And today's time, that concept should not be there. The teacher will teach and you will learn. Only thing, whatever lecture I'm giving to you, I'm giving you the overall view. That's all. If you are interested, you have to read the literature yourself. Then you will learn. Otherwise, you will not learn. After that, if you think that by listening my lecture, you will learn anything, I'm doubt over it. Only thing, you will get some idea. After that, if you are interested, then read the literature. A lot of material is there. Read our papers, and then you will learn it. Okay, this is another resource where we have worked on the other side sort of things, because normally we believe that there are certain type of cancers where the virus is involved, but it has been shown recently that in number of cancers virus is involved. And recently what we did got the information, the lung cancer also have the association with the viruses actually. So what we did actually, we identify the vaccine viruses which are responsible for causing the lung cancer, and then we develop the vaccine candidate, means identify the vaccine candidate which can use to treat these viruses basically, which are responsible for the different type of cancer actually. So you can go through it if you are interested. Then paper says with each server there is one paper. You can read it and you will get the detailed information about these resources. Same way, there is another paper on the prioritization of anti-cancer drugs, because it's not practically possible for you to discover a new drug. But there is another way. There are a number of drugs are already there which are approved for the cancer. Can we prioritize? Can we, let's say, rank these drugs based on the genomic profile of the cancer patient? That's the easiest way. Because these drugs are already clinical approved, it's clinically approved. So it's nobody will challenge you, but you can prioritize for this patient. This is a best drug. Next actually. So what the information we are using here? Genetic information, mutation information, expression information, copy number variation information. And we have demonstrated that for certain cancers, only certain, these are the cancers. Here, this side is a cancer, and this side is a different drugs basically. And it has been shown here that certain drugs work only for certain cancers, some drugs work for the most of the cancers. So that's a concept basically. And we demonstrate here that predicted IC50 value and actual IC50 value, the correlation is between 0.73. And if you combine all these features, this expression, copy number, then up to 0.78. I'm not saying this is a perfection, but it is quite quite good correlation basically. So we can predict the best drug for a given patient. Another problem with the when you are treating the cancer, any cancer, we believe that this is one type of cancer, but this is not true. There are wide range of different type of cancers, even in the pancreatic cancers basically. So here we propose the drugs which can work, which is promiscuous in nature, which can work in the wide range of this pancreatic cancer. And we develop a method where you give the chemical and you will predict whether it can be anti-cancer or not, promises anti-cancer against the pancreatic cancer cell line or not. It can predict it actually. That's the advantage with our servers. You don't need to do anything. It will. You have to put the chemical and it will predict its properties. Now we have developed the methods which can predict the cancer versus normal. This server help you to predict based on the genomic profile. And what we did here is a large scale transcriptomic data actually, because you are getting transcriptomic data from the different sources. So can we have a server which can predict based on the transcriptomic data whether the person is in normal or in the cancer stage basically, in the cancer. So this server will allow you to predict. Same way, this software server help you to predict the stage of the cancer, whether it's early stage or it's a late stage. So gene expression has been used as input and different machine learning techniques have been used. So you can predict it. But as a user, if you are not interested in these things and you are just interested that what is a stage, give the gene expression of certain genes and it will predict it whether it's in the early stage or late stage actually, because the risk is depending on the stage of the cancer. That is for the another cancer. So I'm not going in the all detail each and every one. You can read yourself because recording is already there. You can go through it on my website. Links are there. You can just click them actually. That is again another server for predicting based on the genomic and epigenomic profile based on the early and late stage liver cancer basically. So this is for skin cancer progression, that what is the progress. This is another interesting part, HLA typing. Because that's important. You will see the certain patient are number of people suffer from the cancer. Some people survive long, some people survive less actually. Why it is so? That is based on the immune system. Because MHC or for human it is called HLA, alleles are there and all human being doesn't have the same type of alleles. Some have the certain type of alleles, another have the another type of set of alleles. Some alleles are able to control the cancer, some are not able to control it. So survival will also depend what type of HLA is there actually. Here we try to predict based on the HLA, what type of HLA is there and what is the risk actually of the cancer patients. So here we work only on the skin cancer. This is for the colorectal cancer and based on the 5 proteins actually, 5 protein concentrations actually. So if you have the protein concentration, these are the biomarkers which can be used and we demonstrated this is the best biomarker for predicting the prostate cancer, this colorectal cancer. This is a server, just example of a one server. You put the concentration of the different proteins and it will predict what is the risk actually. So this is just a output page. All right, so there is another server. I'm not going. From the diagnostic point of view, what we did actually, we collect all the cancer-associated peptides found in human body fluid. Human body fluid may be biofluid, maybe blood, maybe saliva, maybe plasma, serum, urine. So we have collected all the information and created the database to develop the different type of biomarkers to detect the cancer, basically. Okay, so peptide based. So far, I talked about the biomarkers, identification, but one of the important part is you want to know how to treat them. So I'm not going, our group is not going in the small molecule based inhibitors or small molecule based drugs. We are in the peptide based drugs. So this server, Cancer PPD, this database is a anti-cancer peptides and proteins basically. So what we did, we collect and compile all anti-cancer peptide and proteins, experimentally validated in compiler here actually. This server allows to predict the anti-cancer peptides. This is for tumor homing peptides. So I'm just trying to cover as soon as possible due to the time. And this is basically for the tumor homing peptides. These are the peptides which have the property that if you give to the body fluid, then it will go and bound with the tumors basically. That's why it is called tumor homing peptide. So we create the database as well as the prediction method, and it can be used for delivering the drug at the tumor site basically, right? This is a recent review we have written. So if you are interested to understand the computer research for identification of cancer biomarkers, then this review will help you. This is published this year itself actually in Briefing in Functional Genomics. So you can go and read this article. You will get the link of all the resources required for that point basically. And it is freely available because it's an editor's choice. So you have no need to worry about the access. You can easily get this paper actually. Thank you very much. I hope I am on the time. I am open for the question.
Monica, unmute yourself, please.
No response from your side. What is going on? I'm not able to hear.
Sir.
Yeah.
Sir,
Now it's okay.
yes, sir. Sir, first question is there any web server or database available for cancer metagenomics?
I'm not remember. However, you can search on the net, but I don't remember at the moment.
Okay, sir. Sir, one more question. Are all the web server and database freely available?
Yes, they are free. All freely available. At least developed in my group, all are freely available.
Okay, sir. Okay. Monica ma'am, are you there?
Okay, if there is no more question, then thank you very much. Thanks a lot.
Yes, sir. Sir, I think we had only two questions. So thank you, sir, for a valuable and very informative talk. And it was always a great experience to listen you. And you always inspire the young minds to make a good research profile. So thank you so much, sir.
Thank you. Thanks a lot. Thank you.
Momento.
Yes, sir. Sir, we are presenting token of appreciation.
Dr. Raghav, this is a token of appreciation from the entire team to you for a wonderful talk and enlightening talk. Thank you so much, sir, once again. Please accept it on behalf of the entire team. Thank you, sir.
Thank you, sir. Thank you so much, sir.
I, Dr. Sushma, on behalf of Department of Zoology, Deshbandhu College, University of Delhi, I extend my sincere thanks to all the respected chair and co-chair for chairing this session and giving us their valuable time for chairing the session. I also extend my gratitude to participants from different universities across the country, audiences, for their active participation in today's virtual international conference on cancer biology. Thank you.
So we have the next speaker for the day, Dr. A.K. Diwan sir from Rajiv Gandhi Cancer Institute. He is the director of Rajiv Gandhi Cancer Institute. And for this, we have Dr. Sadhna Sharma ma'am as a chair to introduce him. And ma'am, please do the honors, ma'am.
Hello, Dr. Diwan, and we welcome you to this conference. And as Manisha has already introduced, that you are the director of Rajiv Gandhi Cancer Institute and Research Center. And for the last, you have an experience of about 38 years. Dr. Diwan has done his MBBS from Maulana Azad Medical College, and in a way, he is an alumni of Delhi University because he did his MS surgery also from RML Hospital with the training at Tata Memorial Hospital, and then his MCH in surgical oncology from Cancer Institute, Adyar, Chennai. Then he did his postdoctoral course in hospital management in from the National Institute of Health and Family Welfare, Delhi. He has been working as a director of Rajiv Gandhi Cancer Institute for the last, from the 1996. He has a specialization in head and neck cancer, and he also has interest in breast cancer surgeries. On an average, he conducts about 120 major and 100 minor surgeries every month and is involved in about 18,000 surgery in course of his career. He is the chief editor of Rajiv Gandhi Cancer Institute monthly newsletter, and he has about 30 articles to his kitty, both in national and international journals, and he has presented more than 350 papers in various medical conferences. With a vision to provide best medicine for the cancer care, he really practices ethically and emphatically. He believes in practicing evidence-based medicine. He's also medical director of a philanthropist organization, Ganga Prem Hospice, which is based in Rayawala, Uttarakhand, which is a palliative care center for terminally ill cancer patients. With so much of experience in cancer surgery and research, we welcome you, sir, to enlighten us with your talk. We welcome you, sir. Please.
Good morning. Can you hear me?
Yes, sir, we can hear you.
Okay, fine. So, good morning, the august audience, friends, faculty members, organizers, and researchers. When I was invited by Dr. Sengar just 2 months back, the golden days of zoology honors in 1975, Hansraj College flashed back in my memory lane. When I did my BSc first year, and my childhood friend is still there in zoology department, Delhi University, and I keep visiting that place, and I love to visit that place, Dr. Rakesh Seth. And thanks and my special gratitude to the organizers, especially Dr. Sengar, for giving me this opportunity. I don't know, today I'm feeling a little nervous. Maybe I'm the odd man out as a surgeon and speaking in front of the basic scientists. Before I introduce my talk, it is actually what is hot, what is not in oncology. It only means what is new in oncology and what we have left in oncology. It is more of a philosophy, and I will try to make it simple and understandable as possible. Even a non-medical man will be able to understand. And let us walk through the journey of oncology. I know when I was a medical student in late 70s, 75 onwards, cancer was a curiosity. And this is what we used to see, all the fungating, ulcerated growths of breast and wounds on the face. And all the medical students used to rush to see how cancer looks like. And we used to have classes on that. But nowadays, things have changed. We detect cancer by screening, by doing a special X-ray of the breast. It is called mammogram. I'm not saying we don't see such cases which are advanced cases. In fact, still we see 2/3 of our cases are advanced cases. But we see more and more of screen detected cases. At that time, we used to see publications which are more clinical and epidemiology. People reporting incidents of one particular cancer and specific treatment of that cancer, maybe surgical technique for that cancer. But in 1980s, we saw technological growth. That time we saw CT scans coming. Good imaging methods came up. And laparoscopy, the endoscopic surgery was introduced. I'm sure everybody must have heard of LAP-CHOLE, laparoscopic cholecystectomy, gallstone removal with the endoscope. So in early 80s, this was introduced, and every surgeon was keen to learn LAP-CHOLE. Even I learned at that time. Then in 1990s came the era of molecular. And we tried to understand the biology of cancer. And I remember when I joined Rajiv Gandhi Cancer Hospital in 96, we were keen to know what is the receptor status of breast cancer, which we now say ER/PR receptor. We started doing routinely in those days because the treatment was based on positive or negative of ER/PR. In last 2 decades, great advances have been made. And what you are listening in last few lectures has actually laid the foundation of personalized medicine. We'll talk about what is personalized medicine. What happened in past, what is present, and how we are heading towards the future of. In olden times, we used to do X-rays. Everybody knew IVP to be done, barium solo, barium meal upper GIT, barium enema to be done. And my teachers insisted that always examine a patient and touch a patient. He'll be satisfied. I call it as a hand scan. That was most important. So all my diagnoses were based on my clinical judgment. That is hand scan. Nowadays, you hear of CT scans, MRIs, PET scans, and even liquid biopsy. Talk about liquid biopsy later. Regarding treatment, surgeons will do the surgery, refer the patient to radiotherapist, or maybe a physician. Radiotherapists used to give chemotherapy also at that time. Even as a surgeon, I have given chemotherapy in those times. So there were no classified medical oncologists or surgical oncologists. All the surgeons did all surgeries. All the physicians gave chemotherapy, radiotherapists gave chemotherapy. And after I have done my job, I don't know what is going to happen to the patient. Patient goes. And I used to say, if there is a problem, you come to me. I have done my job. That was the attitude of clinicians. But now is the era of combined modality. Most of the cancers are treated in a combined way. Combined way means surgery plus radiation plus chemotherapy plus immunotherapy, targeted therapy, and so many things are there. And we all together decide what is best for patients. We do investigations and then decide which medicine. People have talked about personalized medicine, and that has actually given way to personalized medicine. In the past, the surgery used to be quite mutilating, but nowadays we talk of minimally invasive surgery, robotic surgery. We have very good methods of reconstruction of organs. In olden times, we had very few chemotherapy drugs and only cobalt machine with a lot of side effects. But nowadays, we have moved away from cobalt and we have latest technologies of radiation. We have immunotherapy, targeted drugs, and advanced chemotherapy drugs. In the past, the outcome was quite poor, and nowadays we have achieved cure in some of the cancers, especially pediatric cancer, acute lymphatic leukemia, germ cell tumor. And we talk of quality of life, not only duration of life. So there is a shift in paradigm in oncology. And oncology has actually become a complex specialty demanding expertise in various disciplines and exposure to many live entities. As a surgeon, I'm not just supposed to know how to cut. I'm supposed to know when to cut, when to stop my hands, when to give radiation, when to give chemotherapy. So it is basically total care of patients by combining various methods of treatment by multi-professional team with intention of cure or palliation. And emphasis of all this approach is, can we preserve organs? Can we give good quality of life to these patients? And the care is becoming personalized care. But in that bargain, what has happened? In the past, we were low tech and high. Everybody used to examine a patient and touch patient. He used to be satisfied. But nowadays, we are high technology, but low touch. Young doctors, young oncologists, they don't even examine a patient. They'll see the x-rays, they'll read the MRI reports. They'll not even see the x-rays. They'll just read the MRI reports and PET scan and just give their opinion. That is what is not good in long run. The other thing, the other change which has occurred is evidence-based medicine. In the past, I am saying maybe 40 years back or so, the practice was eminence-based. Whatever my boss has said, do the surgery, do the surgery. Because my boss has got more experience and the boss is always right. But nowadays, it is not the eminence of a person. It is the evidence which works. My students question me that if you are doing this surgery or if you are treating the patient this way, where is the evidence? So the treatment is evidence-based, not eminence-based. So the scenario in oncology is changing in diagnostics, in therapeutics, in outcomes, in quality of life issues. I will just show you 2-3 slides in diagnostics. You have heard of CT scans, MRIs, and I'm sure most of you must have heard of PET scan also. Positron emission tomography there. And probably maximum numbers in Delhi, every nook and corner you hear of a PET scan. PET scan combined with a CT scan and even combined with an MRI. PET scan actually enables the collection of both anatomical and biological information. CT scan gives you only anatomical information. Yes, this organ has got this abnormality. But that abnormality, does it show any activity? Has it got any relation with cancer? Or some inflammatory processes going on in that particular area? That is called biological information. That is what we get from PET scan. We may be able to diagnose tumors early. We can precisely localize cancer. We can accurately stage them and plan our treatment precisely, and even monitor the response to our. The other thing which is new is a liquid biopsy. People are quite scared of open biopsy, making a cut, taking a biopsy. And especially if you want to assess the response to chemotherapy or some surgical modality, and you want to see patient is cured, every time you can't take a biopsy. So liquid biopsy means you take a blood sample. And it is presumed that tumors shed DNAs, the dead cells, they shed DNAs, RNAs in blood circulation. If you pick them up and test it, you may be able to detect cancer in simple blood material specimen. So liquid biopsy may prove to be useful for every patient with metastasis or a patient who is under treatment for cancer. Every patient who is in remission or a cancer survivor, and you want to monitor cancer. And if a person is not responding satisfactorily to treatment, and maybe in future, this becomes a screening tool that you do just simple blood test, and you may be able to detect cancer by a simple blood test. You don't need to do a CT scan, PET scan, just a simple test tells you, and it might become a screening tool, especially in people who have got strong families. But today it is not a substitute, right? It is a supplement to the conventional biopsy. So in a nutshell, what will be the basis of future treatment and staging? It will be molecular credentialing, gene sequencing. We'll have gene chips, and the treatment will be personalized treatment. That is the future. And molecular biologists or cancer researchers like you, IT engineers, will dictate treatment, and maybe surgeons may be out of job. Another pictorial presentation of how the things are changing and how what is the future. You see a patient do a biopsy, open biopsy, see it under the microscope, do some other tests, lab tests, treat that patient, and maybe repeat the biopsy if the tumor has disappeared or not, or maybe do some ultrasound, CT scan, that is all. The other way is you have done the biopsy, you do the genotyping, find what is the mutation which has happened. Is that gene change actionable or not? Actionable means you have got any drug or antibody against that genetic material. That is called targeted drugs. Treat the patient with those drugs, or the actionable genes are targeted, and then you can do a PET scan again and see whether the patient has responded or not. But the future is maybe doing a liquid biopsy. Just take a liquid fluid from the body, maybe ascitic fluid, chest fluid, or blood sample, and find out abnormality in the molecular pathways. So many people have talked about the molecular pathways. I'm not going into the details of different pathways, but different protein pathways. And then find the actionable drug against the abnormal molecule. And maybe after treatment, find the circulating tumor cells in blood and try to see whether the person is responding or not. That is the future. So the essence of cancer treatment is the search for cancer cure is difficult, but not hopeless. There are various options of treatment. Surgery, of course, has stood the test of time. For ages, surgery has been the treatment of choice. In last 100 years, radiotherapy has come with or without chemotherapy. Chemotherapy alone, then targeted therapies, immunotherapy, hormone treatment, and the interventional oncology has come. I'll talk about it. But the ultimate aim is personalized treatment. I'll just go one by one, show a few slides for each treatment and how we have changed. And there has been a revolution in evolution of radiotherapy. In the past, when I joined this hospital, even at that time, we had telecobalt, cobalt machines, a lot of side effects. And we used to put blue marks. Even some of the government hospitals even now have cobalt. We used to put on face or on abdomen some marking, blue marking with gentian violet. And the technologist will give radiation in that particular blue area. And there was a lot of scatter radiation, and surrounding areas, normal areas, used to get a lot of. Then came the linear accelerators. And every year now we see new technology. IMRT, IGRT, tomosynthesis, SRS, SBRT, so many therapies, all are actually called teletherapies. And there are various ways of focusing the radiation beams to the organ. And aim of all these is precision, 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 and preserve the organs. Precision means hit only the cancer cell, map them, maybe with an MRI or PET scan, map them, and try to safeguard the normal areas, bombard only the tumor cancers. So the aim is precision and preservation. Precision, treating the diseased area. Preservation means preserve the normal area. And we also have brachytherapy, means contact radiotherapy with needles, with different types of implants, where radiation source is kept next to the diseased area. So what is latest in radiotherapy? You see, you come to know about the radiation effect, whether it has acted or not, only after radiation is over. 6 weeks of treatment, or maybe nowadays proton beam is there, newer techniques are coming up, particle therapies are there. You may be able to deliver the radiation dose early, but you will know the results of radiation only 6 to 8 weeks after completion of treatment. So how can you predict response to radiation? There are ways, research is going on in this. And the other thing which is happening is preserve organs. And these preserved organs should be functional organs. So you have killed the tumor and saved the organ, which is a functional organ. In chemotherapy, we've got newer drugs, newer schedules, newer indications. We have bone marrow transplants. Cure rates have improved. As I said, we have achieved cures in many of the childhood tumors, germ cell tumors. And we use quite frequently NGS, molecular diagnostics. There was some discussion going on that in South Bangalore and Chennai, you have the latest techniques, but I can assure you, Delhi is not behind. We have all the technologies, including next generation sequencing, molecular diagnostics, immunohistochemistry, you name it, and we have the techniques. And we select our treatments, tailor our treatments according to the genetic profiling of these patients. The other thing which is coming up is immunotherapy. All the drugs which you hear ending with the name of MAB means it is immunotherapy. This actually uses genetically modifiable cells or viral particles to stimulate the immune system to destroy cancer cells. The other aspect is oncolytic virotherapy, which uses viral particles that replicate within the cancer cells to cause cell death. still experimental. The other experimental arm is gene transfer, which introduces new genes into the cancer cells or surrounding tissues to cause cell death or slow the growth of cancer. There was some discussion about targeted therapy. What is targeted therapy? As I said, you see, you identify the abnormal molecule and give antibody or something against that molecule so that the cancer cells are killed and normal cells are spared. That is the basis of targeted. And somebody has rightly said these are actually smart bombs versus cluster bombs. Cluster bombing will destroy and kill the normal persons also, civilians also. But if you want to target the terrorists, these are smart bombs, highly precise. And this is what is targeted. Good example is Gleevec for chronic myelin leukemia. So the current therapies exploit the loss of cell cycle control and the genetic instability of cancer cells. But cancer can evolve resistance to treatment. It is not that in every case and every time in a particular person, the drug will work. Cancer can evolve resistance. And new therapies may emerge from our knowledge of cancer. So the whole treatment of personalized medicine is designed to attack particular cancer cells or growth of tumor choked by depriving cancer cells of their blood supply. You can stop their blood supply so that they don't multiply. Or the small molecules to target specific oncoproteins. That is in a nutshell the basis of all this personalized treatment. And understanding of cancer biology, what you have been talking about, leads to rational, tailored medical treatment. And we actually, as a scientist, exploit DNA repair mechanism, cell cycle checkpoints, and apoptotic pathways. A good example is non-small cell lung cancer. You see, we are talking of targeted therapies, and I'm just giving you one example. Person was labeled as stage 4 disease, lung cancer. All this white area is cancerous area, and a bad chest. Normal lung will look like a black area. All this white is diseased area. You have given a tablet with the name of crizotinib in ALK positive non-small cell lung cancer. And within 4 weeks, you see a normal lung. So these drugs targeted therapies can sometimes act as magic bullets. I remember a few years back, differentiated thyroid cancer, when they become resistant to the radioactive iodine therapy, nothing was available. We gave local radiations also, gave high dose of radioactive iodine treatment. In large, 90% of them will do very well, but some of them will develop metastatic disease in lung or bones, and then the outcome is bad. But we have identified the molecular pathways and some of the targetable drugs, especially lenvatinib, we get more than 50% overall response rate in radioactive iodine resistant thyroid cancer. These are all simple tablets. I know they are very expensive. The cost may run in lakhs per month, but they are easily available even in India. So in nutshell, what is latest in systemic therapy? Systemic therapy means which you are giving either orally or injection wise. It includes chemotherapy, immunotherapy, targeted therapy, all that's called systemic therapy. So you have newer and newer drugs. Like very good example is nivolumab, atezolizumab. All these MABs are immunotherapy. MAB is monoclonal antibody. Okay. And anti-cancer vaccines are also available now for some of the cancers like kidney cancers, melanoma. The other new concept which has come up is tissue agnostic therapy. See, in the past, we treated adenocarcinoma of breast differently from the adenocarcinoma of colon, differently from the adenocarcinoma of maybe prostate. But all are adenocarcinomas. Histologically, they're all adenocarcinomas. Only the tissue of origin is different. So what are the molecular changes which have occurred in all these adenocarcinomas? Are they same? Are they different? People have started working on this, that why not treat all the adenocarcinomas of different organs in same way with same drugs? So there is a shift from treating homogeneous organ-based disease to molecularly based heterogeneous diseases. You find the molecular abnormality. Don't treat the organ, treat the particular molecules in whatever organ. So that is called tissue agnostic therapy. And discover drugs with companion molecularly based tests. It means the same thing. So enabling personalized cancer medicine through analysis of gene expression patterns. This is what we are achieving in present era. Being a surgeon, I can't leave the topic without talking of surgery. You may say that what is the role of surgery? It's just remove the tumor or maybe do a biopsy. But it is not so. Surgery is also used for prevention of cancer, especially if there are multiple polyps in colon, or there's a term like MEN, multiple endocrine neoplasia syndrome. And it is surgery is also done not only for primary tumor removal, but sometimes you have treated the patient with radiation chemotherapy. There is residual disease or there is recurrent disease. Surgery may be used. If it has already metastasized, in the past, the thought process was, it is a terminal disease, don't touch. But nowadays, we also do metastatectomy. If there are oligometastases, means fewer metastases. Some of the emergencies in cancer, like bleeding, perforation of intestine, stridor means difficulty in breathing, surgery is quite useful. In pathological fractures, you may fix the fracture and improve the quality of life. You have very good methods of reconstruction, rehabilitation. Some of the simpler methods are there, laserization and HIPEC. I will not go into the details of all this. But most important is sequencing of treatment. Who treats the patient first? Surgery or radiation or chemo? And all the treatments are multidisciplinary. Say oral cancers. Previously, it was the domain of an ENT surgeon, but nowadays, it is not the ENT surgeon who does head and neck surgery. It is the ENT surgeon, head and neck surgeon, surgical oncologist, reconstructive surgeon, maybe a neurosurgeon join hands and decide what is best for patient and how to do it, when to do it, and not to do it. And in the past, the concept was if there is cancer in one organ, just chop it off. Limbs, if there is cancer in the bones, remove it. If it is in the breast, remove the breast. But nowadays, we try to conserve, especially in breast, voice box, neck, thyroid, kidney. There's no need to remove whole kidney. Bone tumors and sphincters in the rectum, infertility can be preserved. I don't know whether time will permit or not, but the minimally invasive surgery has come up in a big way. This is just a small 2 minutes video of robotic surgery for oropharyngeal cancer, throat cancer, early stage disease. And in the past, I remember I used to arrange 4 units of blood and take maybe 6 to 7 hours in doing the surgery, and quite major and costly surgery for some of the throat cancers. But nowadays, you see, these are the robotic arms which are operating, and the surgeon is sitting just 10 feet away from the surgeon, from the patient, and it is all computer assisted arms which are doing the job within your throat. And such surgeries, robotic surgeries, are being commonly performed for urological cancers, especially prostate cancer, gynae cancers, even thoracic tumors, lung cancers, many of the abdominal cancers, and with least morbidity, and you hardly require blood. Patient goes home very early. And such patients can be sent home in 2 days' time, rather than patients being kept in hospital for 8 to 10 days. So this surgery takes about maybe 20 minutes, half an hour, and that's it, no blood loss. The other is limb preserving surgery. If you have osteogenic sarcoma, means bone sarcoma, previously we used to amputate, remove the limb, but nowadays don't remove the limb, give them chemotherapy, do a conservative surgery, means the diseased part of bone is removed, only the diseased part is removed, and replaced with other bone, only the part of the bone is removed, or put a prosthesis. These are prostheses that are available, and person can bend, and limb cancer person can even walk now. So save limbs, save organs. Same is for breast. And yesterday you heard from Dr. Van Chicken. You can tailor your surgery according to the needs. All patients need not have mastectomy. You can do a lesser surgery, do a sentinel node biopsy from the armpit if the tumors are small, but achieve the same results. So there's a new phase of breast cancer management. Do a lumpectomy. Don't remove breast, only remove the lump. Do lesser number of node excision. Even robotic mastectomies are available. You can reconstruct the breast. Radiotherapy need not be given to whole breast, but partial breast irradiation is known. Targeted therapies are available. As the previous speaker talked of, Dr. Chandan talked of trastuzumab or Herceptin in breast cancers. Circulating tumor cells for monitoring response of chemotherapy or targeted therapy in breast cancer. Liquid biopsy, vacuum-assisted biopsy, especially in benign tumors. Gene profiling, genetic counseling, BRCA1 and 2. And I'm sure you must have heard of the Angelina Jolie, who got her both breasts removed and both ovaries removed, probably in 2013 and 15. She never had breast cancer. She had abnormal genes, mutated genes, and she had prophylactic mastectomy and breast reconstruction. But the buzzword in these is early. The other aspect of oncology, which has now started, is interventional oncology, which is done by radiologists, not by surgeon, not by radiotherapist, not by medical oncologists, but the radiologists have come in a big way. They do the biopsies, which are done under ultrasound or CT guidance. They put the catheters. They embolize the vessels of the tumors. They embolize the feeding vessels in liver. They're involved in gene therapy. They can do many procedures which we as surgeons may take more time. Patient might bleed. It'll be more traumatic. They do the procedures with least. And same is nanotechnology. I'm sure as basic scientists, you will know much more than what I picked up from the literature. Nanotechnology, I only know that some of the drugs, paclitaxel, gemcitabine, or the newer versions are available where there is a use of nanotechnology. And nanoparticles have been functionalized with ligands, and even small molecules, DNA, microRNAs have been used. And nano-enabled immunotherapy will be coming soon. And there are plans to deliver and augment radiation with nano. And I have not seen, but nano robots are available for surgical treatment. There are micro grabbers for tissue capture and micro diggers, which dig into the target cells. So the surgery will be possible at cellular, subcellular, and molecular level. Nano robots are powerful navigators. So in nutshell, these are last 3-4 slides. The changes which are coming up are screen detection with liquid biopsy or CAD, that is computerized assisted diagnosis, molecular credentialing, robotic surgery, and we may have even robotic assistants, nanotechnology, immunotherapy, gene therapy, targeted therapy, various types of vaccines. At the moment, very few vaccines are available for prophylaxis, like for CS cervix, for HPV. And most of these so-called vaccines are being used for treatment of melanoma, renal cell carcinoma, and prostate cancer. We will soon have better navigational systems which will work with robots. And I repeat that surgeons will be out of job and molecular biologists and IT managers will decide. But what are the problems in India? We have been talking so much about the latest advances. We have huge volumes. A few people may see it as a problem, and I see it as an opportunity. But there are no screening programs, structured screening programs in India. Loss to follow patients are too high. Once patient is treated by me, he is lost. He'll end up with me or with somebody else if he has problem. A lot of disability and deformity by disease as well as by treatment. All the infrastructure availability issues, affordability issues, training issues are there. And there is urban concentration of most of the facilities. There are no uniform protocols in India. It is unfortunate. There is a lack of awareness. And the overall treatment results are actually 20% poorer than the Western world. We sitting in metros may be able to achieve equivalent result as the Western world. But all over India, it is not so. We have alternative therapies. Everybody wants to treat cancer. Homeopathy, Ayurveda, Botpathy, Nobody will leave. They will give you 110% guarantee for treating cancer and curing. But once it becomes stage 4, then they will say, now you can have a second opinion with somebody else. And it is unfortunate. And the other thing is concept of my patient, ye mera patient, rather than our patient. Together, we need to sit and decide what is best for a particular patient and how will the patient benefit, rather than I treat and I will give you the best treatment. No, maybe there is something different. Some molecule is available I'm not even aware of, and patient might be able to achieve cure with simple oral tablets. And it is happening in lung cancers. So the scene is changing in oncology, and we have moved from single modality treatment to combined modality treatment. But we need to change from the concept of my patient to our patient. Cancer treatment does not end at how. Surgery can be done by anybody, but when not to, and with what result. And I always say, decisions are more important than incisions of surgery or precisions of radiotherapy. And we need to move towards personalized care. And the interventional oncology is coming in a big way. And we must focus on prevention of cancer and vaccination of cancer. And soon the treatment of cancer will be non-surgical and it will be personalized treatment. And my appeal to all the researchers is research. Do not research. Whatever is done, you start saying, I will do the same thing. Bring lab to the bedside and be the backbone of oncology. And I can assure you, you are the future. Thank you.
Thank you, sir, for your enlightening lecture. We came to know about whatever is latest in cancer diagnosis and treatment, as well as future approaches in surgery, radiotherapy, even systemic chemo and immunotherapies. All of it is, which you say, is aimed towards personalized treatment with targeted therapy and, of course, the anti-cancer vaccines. With your scan, your personalized care and practice of evidence-based multimodal conservative approach, and of course our patient approach, we believe we are in good hands as far as our health is concerned. And whatever you say, surgeons will never go out in India, especially where personalized touch is required. You will be always there. It is very nice and very enlightening to hear from you. Thank you, sir.
Thank you so much, sir. It was a very wonderful lecture, sir. It was an eye-opener for all of us. It was truly amazing, sir. Truly amazing.
Yeah, really hearing it from the doctor itself was very nice. Yeah,
exactly, ma'am. And we came to know, sir, so many things about it, and we were, we just read and we just see it from the news and everywhere. But coming straight from you is something different, sir. So it was a wonderful session, sir. Not a lecture like this we've heard ever. It was so good, sir. So good. Thank you so much, sir. So there's a token of appreciation for you, sir. Thank you so very much, sir. It was a wonderful, amazing lecture. Did not waver our attention a bit, even for a second. It was we were stuck up to the screen and we were just listening to you. Thank you, sir. Thank you so much, sir. Thank you for accepting our invite. It is a great honor, sir. It's a great honor. It's a great honor for us.
Thank you.
Thank you so much, sir. Thank you so much.
Manisha, can we have some questions also?
Yes, ma'am, we will. We will, ma'am.
Yeah, first of all, I would like to ask one question. You talked about Angelina Jolie effect. Like personally, we never like we are being studied cancer and did our doctorate in cancer also. I never believed that doing this kind of things which Angelina Jolie actually did will really help her in not getting cancer. What is your opinion?
Yes. You see, we now know that if BRCA1 and 2, they are mutated, the risk of developing breast cancer is to the tune of 60%. And risk of developing ovarian cancer maybe 40 to 50%. So people are scared. And they want to get rid of possible source of trouble. And they also know that there is reconstructive facility available. So they say, why not remove the troubleshooter, but aesthetically have the same thing. Aesthetically have the same thing means breast mount can be given. Nipple can be spared. Silicone prostheses are available, which can be put inside. So the humanity remains. But the risk of cancer goes. I think the pain of cancer, only a cancer survivor or the kith and kin of cancer patient knows. Angelina Jolie's mother had cancer, breast cancer. She never had cancer. But she was all the time scared that she might develop after seeing that she has mutated genes. Previously, I was also against such surgeries, but I started doing all that, knowing the fact that people can benefit from such prophylactic surgery. So we also at Rajiv Gandhi Cancer Hospital explain the patient what are the benefits and what are the negative effects. And we have a department of genetic counseling who will tell them that whether you should have the investigation at all or not, at what age, how will you benefit, what are the options available with you, can you be put under screening protocol or not, how frequently you should have screening. Such people can have MRI frequently every 1 year. So all these are there. If the gene is mutated, you can do either the follow-up or do prophylactics. Benefits. Yes.
Okay, sir. Thank you. There was one or two more questions regarding use of long coding RNA instead of along with microRNA in doing liquid biopsy. Do you have any idea about it?
I think the wrong person who answered this question. The basic scientists are sitting amongst the audience. So, which RNA or which DNA fragment can be detected in which cancer? They're all different. Where what will help? Probably I may not be able to tell you with research.
So, of course, you have talked about nanoparticles being used for some kind of chemotherapy, but does it involve gene therapy also, or people are doing such work?
No, they are all experimental. They are not in common use. But nanoparticle being used with paclitaxel, and the efficacy is supposed to be much better. Paclitaxel in the past used to cause a lot of side effects, but the side effects have definitely reduced with the nanopaclitaxel. So I think the future is with these genes.
Okay, so with this, I again thank you for giving such an informative and enlightening talk. We really got benefited from your talk.
Thank you. Thank you.
Thank you, sir. Thank you so much, sir. Thank you for sparing your time, sir, from a very
Thank you.
busy schedule. And I think you are sitting outside the
I'm
operation
sitting
theater.
inside the operation theater.
Yeah,
Oh my
it's
God,
heat.
sir. Oh my God. Oh, that's something really big, sir. Thank you so much, sir, for
Okay, thank
taking
you.
out your
Welcome.
precious
Bye.
time,
Bye.
sir. Thank you, sir. Thank you so much, sir.
Thank you, Manisha ma'am.
Thank you.
I, Dr. Gauri, on behalf of the organizing committee, International Conference, extend my sincere gratitude to Dr. Devan sir, Professor Sadhna ma'am, and Dr. Pratibha for an enriching session. We all will be breaking off for lunch now and will reassemble at 1:10 sharp for parallel oral and poster presentations in the respective Zoom rooms. After joining the Zoom rooms, please look for the break room options on the main screen and choose the room. In case of any problem, please let us know. We'll be meeting back at 1:30 and hopefully everyone has filled in the feedback forms also. Thank you.
Thank you, Manisha, for your invitation. Thanks a lot.
Thank you so much, ma'am. Thank you so much, ma'am. Thank you for sparing your time, ma'am. Thanks a ton.
Pleasure
We'll
is
be
mine.
I pardon. We will be meeting back at 1:10 after lunch for the parallel sessions.
Thank you, Jha sir. Sorry, sir. We will unmute you. We'll try unmute kar dein Jha sir ko. Sir, thank you. Just a minute, sir. We'll just unmute 1 minute. So you are always, motivating us. You're always there supporting us, sir. We are really indebted. Every time we organize something, your presence is always, awaited now. We wait for your presence, sir. We really wait for your presence.
I thank you, Manisha, and entire team. Good opportunity you have given to me. So,
Thank you, sir.
information, first hand information from doctor and researcher. Very, very good. Thank you.
Thank you, sir. Thank you so much. Thank you, sir, for sparing your time, sir. You really take out time. It's a big honor for us, sir. Thank you so much, sir. Thank you.
Manisha, do you think that I should join also poster and that thing? Is it interesting for me?
Sir, they're very good. Yes, sir. They were very good yesterday also. They were superb. Very good poster presentations and oral presentations were there, sir.
Okay,
You should listen to them also, sir.
Okay, thank you.
Thank you, sir. Thank you, sir. Thank you, sir. So we'll join back. Okay. I'll leave.
On behalf of the organizing committee, there's a few announcement, emergency. The Zoom room will break out in 35 seconds, so we can come back again, all right? So stay put. A small technical glitch. In 35 seconds or 30 seconds, the Zoom room is going to break out, but we'll bring it out again. We'll restore it again. So kindly stay put. Good afternoon, everyone. The program will be starting in 20 minutes. So those who are in the main room can have some tea and come back in 20 minutes sharp. Thank you.
Good evening, everyone. Am I audible? May I request Kulbhushan or the organizers to please let me know if I'm audible?
Yes, Monica, you are audible.
Okay, thank you, ma'am.
And can you switch on your video so that I can confirm whether you're...
I am there, ma'am.
Yeah,
I'm
video.
here only. I'm here only. My video is on.
Okay, so here is my friend. Hello, Avdesh. Hi, nice to see you after ages. Just a minute, 1 minute, 1 minute. Sir, Avdesh ko unmute karo, host bana do Avdesh ko. Just a minute, just 1 minute, 1 minute, 1 minute. One minute. Avdesh, please unmute yourself. Please yes.
Right. Yeah, it's good
Hi.
to see you too.
It's good to see you. You're a big man now.
No, I'm not.
So many awards and accolades to your credit. Oh my God, it's a 22-page CV. So many publications. Oh my God, it's too good. So we'll just start in around 10 minutes.
Okay.
I was able to see your screen too.
You're able to see it?
Yes, I was able to see it. I'll just confirm you. Yes, I am.
Okay, let me put it on show. Okay.
We'll have to wait for exact 5:30. Is it okay? I'll just make a call or one call or two. I'll just get back in a second. Okay, I'll just make 1-2 calls. Okay. Oh, I'll have to quit. Mm-hmm. So, hi, Avdesh. You'll see my sister on the screen. Deepthi, Deepthi, say hello, and you will be surprised to see Sunita, right? She's right there. Pura gang mil gaya hamara sara.
Good to
So
see
we
you.
are all ganging up here. So Deepthi is also in US, and Abhi, I'll just introduce her. एक बार 5-7 minute हो जाए और, and then we'll just have a student gathering too. We just finished with our oral and poster presentations. Just finished around 10 minutes back. So maybe the students are very tired, so they haven't been able to join. And at
Yeah,
least
certainly.
Sunita, आपको भी मैं co-host बना देती हूँ. Talk to Avdesh in the meantime. Sunita Blakely को co-host बना दे. Done. Sunita, you can unmute yourself now.
Yeah. How are you?
Dr. Jetli, how are you? Long
Fine.
time no see.
I know. I'm seeing a transition from a fully hair grown person to.
But you still look the same.
I don't know, but I can see a full transformation in you.
Really? Yeah, I lost my hair for sure.
My father used to tell me, all intelligent people have very small hair. So
Right.
you can now come into a category of highly intelligent because you have lost your hair.
I don't know about that. Intelligent people would know how to take care of their hair.
That's not in your control. Hi, Sunita and Avdesh. I'm Deepthi. Nice to meet you.
Hi,
Nice to meet
how are you?
on Zoom.
Yeah, this is my real sister, the only sister I have. She's the only sibling. She's also there in Chicago, and she's also a doctor by profession. So she shifted after she got married. Yeah. She's an infectious disease specialist in Loyola
okay.
Medical University. Yes.
I'll have to be careful now.
Right.
No, you don't. I was
So
just
she
going.
is-
It's
Yeah.
very.
Sorry, what was that?
I
Yeah,
said
I was just saying your topic seems to be very attractive. Genomic epidemiology.
Let's hope it's not just the topic.
And then you train at WashU, and my son goes there. He just started.
Oh, okay. Okay.
Yeah. And I know it's supposed to be #1 in genetics in the country.
Yeah, I mean, I don't know about the ranks, but it is definitely a good place.
Yeah, yeah. Well, he picked WashU over the IVs, so it has to be good.
Yeah, it had better be good.
Yeah.
Yeah.
But this year was crazy, the admissions and everything. It was a nightmare. But anyways, I'm so happy we are past that. We basically applied without even like seeing a single school or a single school, and we just applied like to several places wherever he wanted to. And once he got in, we toured a few places. But this is what he liked the most out of all. So the campus is really nice.
Yeah, the undergraduate campus is really good. The Danforth campus, it's really nice.
Yeah, yeah.
I only went there once or twice to hear some lectures, but it's really nice. Overall, I think Louis is a very good city.
Yeah.
Yeah.
So we're waiting for others to join in, Manisha?
Yes, because they just finished with their oral. They waited for around 2 hours for their chance. So they are just going to be back. Maybe yes, we have right now over 100. So can we start 108 hai abhi?
It's a good number, spiritually. 108. Right to begin.
Holy number. Yes, let's begin then. So my co-chair is there. Monica, are you there? Monica, can you switch on your video and audio and just let me know whether you're there? Monica? Okay, doesn't matter. She has to do the half of the job, the rest of the job after your lecture. So today I'm really overwhelmed to see my friend here as a speaker, come as a speaker. And I'm really very proud of you. You are such an eminent personality. And for introducing you, I have here Dr. Deepti Chauhan, my sibling sister, and also a doctor by profession. She is an infectious disease specialist in the Loyola University, Medical University, Chicago, Stritz School of Medicine. She did her MS from US, and your co-chair will be Dr. Monica Sharma from Miranda House. She is also from Department of Zoology, University of Delhi. So Deepti, you can introduce Professor Kalia. Professor.
Hi, everybody. We have the honor of having Professor Avdesh Kalia, and we're really, really excited and, looking forward to hearing him. He is a professor, School of Health Professions, Division of Academics Affairs, and University of Texas, MD Anderson Cancer Center, Houston, Texas, which is a very, very renowned cancer institute in the US. He's also associate graduate program director at the MD Anderson Cancer Institute. He has other a lot. He has a lot of other appointments. He's associate editor, infectious diseases, frontiers in microbiology. He's also associate editor, infectious diseases, surveillance, prevention, and treatment, frontiers in public health. He's the chair program committee. He was the chair program committee in the 138th annual conference of Association of Clinical Scientists, Houston, 2017 to 18. Member of document developer committee on molecular methods of genotyping and strain typing of infectious organisms. He's a very prominent figure, and he has also a lot of academic appointments. He's a professor, School of Health Professions, Division of Education and Training, University of Texas, Anderson Cancer Center in Houston, Texas, associate professor, Department of School of Health Professions, assistant professor, Department of School of Health Profession, Division of Academic Affairs, senior VP office, University of Texas, MD Anderson Center. He is also associate professor, Department of Microbiology and Immunology, Louisville, and assistant professor, Department of Biology, University of Louisville, Kentucky, assistant professor, Department of Microbiology and Immunology, and he is also research associate, Department of Molecular Microbiology, Washington University School of Medicine, St. Louis, postdoc fellow, School of Public Health and Department of Ecology and Evolutionary Biology, senior research fellow, Department of Microbiology, All India Institute of Medical Sciences, from 1995 to 1999. So he actually did his training, and let me just get this right. There's so much. Just a second.
He has a 22-page CV. Oh my God, that's too much. We can just discuss about his awards and honors, because he has a number of publications.
His master's and bachelor's in zoology from University of Delhi, and then postgraduate training in population genetics and molecular evolution, microbial pathogenesis at Yale University. And he also was a research associate, evolutionary genetics and genomics, Washington University of Medicine, St. Louis. Then he is board certified MB, ASAP, Molecular Biology, American Society of Clinical Pathology to present. He has also earned several awards and honors. The list is like endless here, but let's see. Inducted to MD Anderson Academy of Health and Science Education 2021, University of Texas System Reagents Outstanding Teaching Award 2020, Faculty Inductee Alpha ETA National Scholastic Honors Society 2018, Faculty Educator of Month, MD Anderson Cancer Center, September 2016, inducted Fellow of Association of Clinical Scientists, November 2015, Red and Black Mentor Recognition, University of Louisville 2010, Favorite Faculty Award, University of Louisville 2008, Ralph Paul Junior Faculty Enhancement Award, Oak Ridge Association of Universities in 2006, Favorite Faculty Award, University of Louisville 2006, James Hudson Brown Alexander Brown Postdoc Fellowship, Yale Award, Yale University, 2000 Emerging Infectious Disease Postdoc Fellowship, CDC Atlanta, Georgia 1999, Special Student Honor Oration Award, American Society of Microbiology in 1998, Young Investigator Award, International Society of Chemotherapy 1997, Junior Research Fellow, National Eligibility Test, National Fellowship conferred by Council of Scientific and Industrial Research, Government of India 1993, and Junior Research Fellowship via the Graduate Aptitude Test in Engineering conducted by University of Grants Commission, University, Government of India in 1993. So we have the honor of, he's earned several awards and accolades, and we have the honor of having him and talk about genomic, let's see. Sorry, I have like too much info here. So he'll be talking on genomic epidemiology of bloodstream infections in leukemia patients. And Dr. Kiran and Dr. Rahul Jambal are his technical team. And Dr. Gajen Singh and student volunteer Samya is also here along with us. So thank you so much, Avdesh, for coming in and enlightening us and all our team and students.
Thank you so much. So we'll not keep him waiting. Go ahead, Avdesh. Thank you so much for accepting our invite. Bless you.
Well, thank you, Doc, very much.
We are eagerly waiting for your talk. Yes, thank you.
All right, and Dr. Sanger, thank you very much for the kind introduction, which basically reminds me, listening about myself, I used to be cooler than what I am right now. So let's hope that the talk goes. Okay, so it's a privilege to be here, back presenting at the Delhi University from where I have graduated. In particular, I did my bachelor's actually from Deshbandhu College. So it's a double privilege for me to be back and give this talk. So for the last 5 or 7 years, I'm trained as a clinical microbiologist, and I've done a lot of my research in evolutionary biology, molecular evolution of infectious diseases. But over the last 5-7 years, my interests have maneuvered maneuvered themselves towards understanding the biology of symbionts in the context of cancer patients. So when symbionts get the opportunity to cause infections in immunocompromised hosts, we typically like to call them as pathobionts. So what you see here on your slide on the left of the picture is a biofilm, which is a community of bacteria getting together and becoming virtually impenetrable, being in process on a catheter surface. And in this particular form, it has two devastating consequences. One, that it becomes impenetrable to most antibiotics, and two, cells within this biofilm tend to be very, very antibiotic resistant. Oftentimes, these infections caused by bacteria, such as the biofilm causing bacteria that you see here, Staph epidermidis, tend to have adverse consequences on patient outcome, especially among cancer patients. So why cancer patients are so susceptible to infections, in part can be explained from the disruption of physical barriers and defenses in the host. So on your left, what you see is a list of defenses that are overcome in a cancer patient, and specific lesions that result from the type of defense being breached. So for example, breaks in the skin make the patient quite susceptible to cancer patient, quite susceptible to infection. Of particular note are two particular categories. One, of splenic clearance of microorganisms as a result of splenectomy, which is often the course of action in advanced cases of CML and chronic myelogenous leukemia, for example, and hairy cell leukemia, where the spleen is removed and the patient becomes severely immunocompromised even after the leukemia has resolved itself. The second category that is of special interest to us is the category where phagocytosis is disrupted, resulting in lack of granulocytes in the blood, or more specifically, neutrophils in the blood. For the students in the audience, neutrophil who may not be aware, neutrophils are the first line of defense or innate defense in the human body. So with falling numbers of neutropenia, the neutropenia can result typically from the cytostatic therapy that is implemented during the induction phases of treating leukemia. And of course, you have other causes like a lack of antibody production and lack of T cells. So when it comes to leukemias themselves also inherently display immunological deficiencies. So for example, multiple myeloma patients typically suffer from hypogammaglobulinemia, and that renders them very susceptible to certain kinds of symbionts or pathogens. A similar case is seen with chronic lymphocytic leukemia and AML and ALL, or acute myeloid leukemia or lymphocytic leukemia. Granulocytopenia or neutropenia happens to be the typical cause. Of note is the idea that glucocorticoid chemotherapy that is administered in cases of non-Hodgkin's lymphoma and acute lymphocytic leukemia sometimes results in T cell and B cell dysfunction, and that the take-home point here is that the patients have inbuilt immunodeficiencies, sometimes triggered by chemotherapy in the induction phases, that renders them very susceptible to infections. And what kinds of infectious organisms are typically isolated from leukemia patients? The list is pretty exhaustive, but some bacteria are very commonly isolated. So for example, Staphylococcus epidermidis in particular, which I'll be talking about in detail, which has over the recent years, become a very common isolate from bloodstream infections in leukemia patients, Staph aureus, and the list goes on and on and on. Now, there was a time back in the 50s and back in the 60s and in the 80s, when a majority of deaths that were resulting, especially in acute leukemias, were essentially because of infectious because of infections rather than the leukemia itself. In fact, several studies that examine autopsy records back in the 70s and 80s established bloodstream infections as the major cause of mortality in leukemia patients and not leukemia itself, which is very, very striking. And of course, with improvements in empirical antibiotic therapy, early administration of empirical antibiotic therapy, and we have somewhat managed to improve patient outcomes in leukemia patients, but not that much. So here's an example of a study that was done about 15 years ago, continues to be a landmark study which basically displays that the risk of bacteremia varies according to the type of malignancy. So in this graph, for example, what you see is different graphs here indicate different diseases, these different line forms. And over a period of 330 days following the diagnosis of cancer, the patients continue to remain susceptible to infection, but certain types of leukemias are more susceptible than other types of leukemia. So for example, here, acute myeloid leukemia, patients have a high susceptibility towards developing a bloodstream infection or bacteremia more formally, whereas patients with Hodgkin disease seem to have a lesser probability, even though they do see an increase in susceptibility as time goes on. So in a nutshell, the risk of bacteremia or bloodstream infection varies according to the type of malignancy. And over here on your right, what you see is the type of bacteremia, which essentially means characterized by the nature of the pathogen itself. So Staph aureus, coagulase negative staphylococci tend to be very commonly isolated in certain types of leukemia. So what you will notice is that even as the risk of malignancy, and even as the risk of bacteremia varies according to the underlying disorder, the risk of mortality, which is listed over here, varies according to the type of bacteremia. What that means is that certain types of bacteremia caused by certain types of pathogen tend to have a higher or a lower mortality. As an example, if you would look here in cases where bacteremia has a polymicrobial nature, you have a raw mortality rate of about 50% and an adjusted 30-day mortality rate of 44%. Whereas if you compare that to beta hemolytic streptococci here, the rate of mortality the adjusted rate of mortality is 38% and the raw mortality rate is about 8%. So the take-home point here is that the risk of bacteremia varies according to the type of malignancy, but the risk of mortality varies according to the type of bacteremia. Now, Staphylococcus epidermidis, which belongs to the category of coagulase negative staphylococci, have been emerging as a major cause of bacteremia in a variety of leukemia patients, but especially as in acute myelogenous leukemia patients. And this is what I'm going to focus today, one study that we did with our colleagues here at MD Anderson. And just to give the students who may not be aware of Staphylococcus or microbiology in general, a quick background on Staphylococcus as a cause of bacteremia. It's one of the most commonly isolated nosocomial pathogens from high dependency patients. And what that basically means is that patients who are dependent on interventions and have typically long hospital stays. The danger from Staph epidermidis, which is a part of the normal flora for healthy humans, lives on your skin and therefore the name epidermidis. Many clinical strains of S. epidermidis produce biofilms, which is what you see over here on a valve surface, which facilitates colonization of percutaneous medical devices. So devices such as indwelling catheters or pacemakers are especially susceptible to being colonized by Staph epidermidis. and that can have devastating consequences for the patient. The other key feature of S. epidermidis bacteremia is that it is indolent, which basically means it typically does not produce features such as fever and pain, and therefore management of a leukemia patient who has staph bacteremia but no symptoms becomes very, very difficult. And the last key point that you need to keep in mind is that most community-based strains of Staphylococci tend to be susceptible to antibiotics, including methicillin, which is a very commonly used antibiotic. But most hospital strains tend to be multi-drug resistant and are associated with very poor patient outcomes, especially in cancer patients. So the drug of choice in staph, that is MDR, and is resistant to methicillin, which is where it is called as MRSE, or I'm trying to write it on this so you can see my hands are a little bit shaky. And this is before I have had any coffee. So methicillin resistant staph epidermidis, the drug of choice there then remains, either becomes vancomycin or glycopeptides or oxazolidinones like linozolid. Now, in oncology patients, as part of their treatment protocol, whether empirical or whether the patient comes into the clinic with symptomatic, is the use of linozolid. And linozolid is basically an antibiotic, which, as you see over here, is targets the ribosyl peptidyl transferase center, which is right over here in the 23S5S ribosome complex, and commonly used to treat gram-positive bacteria that are resistant to many other antibiotics, as I mentioned, like methicillin resistant staph epidermidis. Resistance to linozolid has been on increase over the last 2 decades. It has emerged in several countries, and it arises principally as a consequence of 2 pathways. One is mutational acquisition, where mutations arise in the 23S ribosomal RNA and other ribosomal genes, such as the L3, L4 proteins, and via modification of the 23S ribosomal RNA by the methyltransferase called as chloramphenicol and florifenicol resistance gene. So if we take a deeper look at how the action of linozolid, linozolid basically sits in the PTC, the ribosyl peptidyl transferase center, and inhibits translation as a consequence of this binding. What the CFR methylate and any mutation in this binding pocket can render the drug ineffective, and the bacteria acquire resistance, develop resistance. In B, what you see is the action of CFR gene, which causes methylation of the A2503 position, as a result, resulting in a steric clash or alteration in the binding pocket of linozolid to the 23S5S complex, together the PTC. And as a consequence of this steric clash, linozolid is no longer able to bind to this complex, and therefore is unable to inhibit translation. And that's how the bacteria continue to live and acquire resistance. Now, 2 key things I want you to keep in mind. The 2 forms, the mutational form, where de novo mutations arise and cause resistance, and the CFR mediated resistance typically tends to be acquired resistance, which means it is mobilizable. It can move from one cell to another cell, or one clone of the bacterium to another clone of the bacterium. Now, in this context, here's where our study began, when one of our clinical collaborators, Dr. Sam Shelburne at MD Anderson, looked at the data from 2007 onwards and noticed that linozolid resistance was increasing among patients, among leukemia patients. very, very significantly. So here what you have is the year during which we tracked the data, and mostly from 2013 to 2016. And here is the percentage of S. epidermidis bacteremia isolates that were resistant to linazolid. So you can clearly see that with starting with 2014, somewhere between 2013 and 2014, some event led to a drastic increase in the emergence of linazolid resistance. What he also noticed was in figure B is that since 2007, there was also a dramatic increase in the use of linazolid by the patients and prescription of linazolid by the oncologists, and especially starting at the year 2007. So here, this is a regression analysis where we're looking at the data before June of 2010, and this is after June 2010. And in both cases, you can see that, especially 2007 and 2008, a dramatic jump in the use of linazolid over time. So the natural question that we asked in the lab was, does the increase in linazolid resistance that we are seeing here, does this correlate or does this reflect the clones of S. epidermidis adapting to linazolid exposure? In other words, to keep things simple, more use of linazolid, is that resulting in more or increased resistance to Staphylococcus epidermidis? So to answer that question, I had a very talented graduate student, Shichi Li, who took on the challenge of answering this question. And she broke this question down into two simpler questions. If we have to answer the previous question, which was, does the increase in linazolid reflect the exposure to linazolid? She asked if linazolid resistance in S. epidermidis was widespread, and then further broke that question down into what genotypes or clones of S. epidermidis are recovered from patients, and if linazolid resistance was widespread or restricted to certain clones. Now, those in the audience who have little exposure to microbiology and to students in general, the canonical thinking is that bacteria divide by binary fission, and therefore they result in two identical daughter cells. So each daughter cell should be a clone of each other. And in bacteria, however, two evolutionary processes contribute to generation of diversity within a species, giving rise to discrete population structure within each species. And that is sort of modeled over here, where a bacterial cell divides from one ancestral cell into two daughter cells, and the process of binary fission continues. Over time, which is modeled here from past to present, replication errors cause mutation accumulation, as you can see with these different colored cells. And recombination in deep phylogenetic linkages can also create similar diversity and can actually have an enhanced diversity, such that if you were to sample a bacterial population over in a present time, you will see multiple different kinds of clones circulating in the human population. Now, in order to answer these questions, what genotypes of S. epidermidis are recovered, and if linazolid resistance is widespread or restricted to certain clones, we need to undertake what is called as bacterial genotyping. So the question then arises is, how do we determine bacterial genotypes? And just for the benefit of the students and to keep things less complicated, there are clinical guidelines available. For example, just last month, we published a clinical guideline for the Clinical Lab Science Institute here. It just came out just last month. And one starts to look at finding the differences between members or individual isolates of a species, one immediately realized that the contemporary process of genotyping a given bacterial species involves two phases. If a laboratory phase, which involves culture of bacterial cells, which involves extraction of DNA and which involves sequence data production by using next generation sequencing, and a computational phase, which involves the use of several different tools, considerations for quality control and of analytical choices, and an analytical pipeline. Now, to keep things very, very simple, I'm not going to go into the details of any of these methods, but for the benefit of students, the analytical pipeline can be subdivided into 4 phases, where we take the raw sequence data and use it directly to interrogate public databases, such as those that have multi-locus sequence typing information. And I'll come back to MLST in a little bit. AMR databases, which is antimicrobial resistance databases or virulence factor databases using distinct tool. And after that, you have 3 distinct steps. The first step is assessing sequence quality, where you look at the quality of the data. The second step is to look at data processing. And data processing itself can take 2 different forms. One, it can involve reference-based genome mapping. That means you have a reference, such as the reference human genome. So any other human that you sequence, and if you want to determine differences between one human and others, and the reference, you generally will take your sequences and align them in a process called as mapping to the reference human genome, and then determine what the differences are. That's called reference-based genome mapping. And the second option that we typically take is that of de novo genome assembly. So we will take all these sequences and come up with some kind of a computational method that will allow us to de novo assemble a complete or a draft genome assembly. And then that can be used for widespread purposes downstream, in fact, such as to answer the questions that Shichi was asking. And the third phase is the genotyping, using the reference-based method or the de novo assembly method, genotyping the bacterial strain by looking for single nucleotide polymorphisms in the population, and by assessing the similarity quotient between different strains of the same species. So as I promised, I will talk about MLST a little bit, because it is still the gold standard of genotyping as far as bacterial species are concerned. And this process essentially involves using information for internal fragments of 7 different genes, which are shown to you in these different colors. So 1, 2, 3, 4, 5, 6, 7. And if you can't see it clearly, I'll zoom it in a little bit. And then using the sequence information to develop an allelic profile. So you have 7 different genes. Each gene, each strain will have some kind of an allelic variant for each of these genes. And then using the combination of these allelic variants that you see here, inferring a genotype. Now, 2 definitions are important in this case. The definition of sequence type. Now, the combination of the genotype resulting from the combination of these 7 gene alleles of these 7 genes is referred to as a sequence type. So it's a unique combination of alleles from all 7 genes. And when we look at the evolution of different sequence types, the collection of sequence types that are similar to a central sequence type in 4 or more loci establishes a clonal complex. So just to give you an example, so if this is an ST, if this ST is related to another ST by one mutational, but different in one mutational step, this is called as a single locus variant. And if this ST has given rise to another variant, which differs in another allele, this is called as a double locus variant, or a DLV. And finally, if this gives rise to another descendant which differs in one more variant, this is referred to as a is as a triple locus variant. So this is what we mean by a clonal complex. And this is the central ST, and this then becomes the clonal complex. Now, this is important for me. This is important to understand because when we look at the global population of Staph epidermidis, what we see is we can generate maps like these. These are called as minimum spanning trees. And what we observe is that 74% of global isolates, and this includes thousands and thousands of different isolates, comprise a clonal complex 2, which is shown to you over here. And so this is clonal complex 2. This is comprised of a central ST, which is a lineage called as ST31, ST2, and this represents 31% of global isolates. So in other words, the take-home message from here is that we can organize the multilocus information into a minimum spanning tree maps that look like this, where you have a central ST and its descendants mapped out. And sometimes the descendants also have their own descendants. So this together represents a clonal complex, and the most common widespread clonal complex in at this point is the ST2 clonal complex. So when we did similar analysis on 176 S. epidermidis strains, so Shichi took 176 isolates from leukemia patients and determined their genome sequences and performed de novo genome assemblies, and then did the analysis that is shown to you on the screen here. What we found here was something that looked like this. So we have 176 strains, most of which could be organized into 2 STs. And very surprisingly, what we found was that we did not see a preponderance of ST2, which is globally dominating. But we saw that most leukemia patients at our hospital had a clonal complex of CC5 because the central ST was ST5. And then it had these different descendants, some single locus variants, some double locus variants, and some triple locus variants. And you'll also notice that both in ST5 and ST2, nearly half of the strains have a red color in ST5 and half have a yellow color. Those that are in yellow color are those that were determined to be linezolid resistance, which is what you see on the key provided to you here. So overall, about 22% of ST5 isolates were linezolid resistance, which is a strikingly high amount when one looks at linezolid resistance rates globally. We also saw some resistance to linezolid, one or 2 strains resistant to linezolid in ST2 and in ST22. So the question then became, was the strains that are resistant to linezolid, are these strains adapting to exposure to linezolid, and were these strains to begin with susceptible to linezolid, and then when exposed to linezolid, did they develop resistance or adapt to the presence of linezolid, or does the emergence of linezolid resistance in our population reflect a spread or transmission or dissemination of a single resistant clone? So overall in our data, majority of linezolid resistance was confined to ST5 and the closely related ST6, one isolate, and very few isolates elsewhere in the population. Now, to answer this question, what we needed to do was perform whole genome sequence analysis. And we wanted to analyze all 176 strains, the genomes of all 176 strains. So the de novo assembly approach for all would clearly not work. So we took the approach for reference-based genome mapping. But when we looked in NCBI databases and elsewhere, there was not a single reference genome available for an ST5 organism, and multiple references were available for ST2. So the first thing Shichi had to do was to sequence a ST5 S. epidermidis isolate that was resistant to linezolid, and we did just that. And what she identified, so here is the sequence of the strain, and the strain basically has a 2.5 megabase genome, has conventional features of Staph Staphylococcus genus, including the Staphylococcus chromosomal cassette, which has the methicillin resistance gene MEC, and also is sometimes useful in typing of staph isolates. It has some phage insertions, but most interestingly, the strain had 3 different plasmids. And plasmid PMB151A, which is a nearly 50 kilobase plasmid, is something that I'm going to focus on in a couple of minutes, because this plasmid contains the CFR gene. So you have the gene that can confer resistance to linozolid when present by methylating the peptidyl transfer component of the 23S5S ribosomal RNA complex born on a plasmid, and you can ask what could go wrong. Of course, the danger always is that it's plasmid borne, so it can rapidly spread through the population. Now, the next step we did, since we are establishing a good reference genome for further characterization, was Shichi compared the ST5 strain with several other ST strains from Staph epidermidis. And what she found was that differences between the ST5 strain here and among other strains that are shown to you here in these concentric circles. So just a quick walkthrough for students who may not be intimately familiar with these kinds of analysis. So the inner concentric rings, that the red color ring that you see over here, that's the MB151 chromosome. And each concentric ring in different color is basically a different genome, which we are comparing to the MB151 genome using a very simple technique called genome blast, which many of you might have heard of. And what we find is that the differences are essentially localized to the SCC cassette, to sites where you have phage insertions. So we found 2 phages, 1, 2, and to sites where you have a pathogenicity island, which in bacterial pathogens tend to be quite mobile, and in places where you have transposon insertions. And rather strikingly, we also had a case of chromosomal insertion of a beta-lactamase gene, upon which I won't dwell any further. So lot of conservation, as you see in solid colors, but also a lot of variation between different genomes, as you see in these white segments here. White segments here are denoting essentially lack of similarity. Now here's a detailed look at the CFR plasmid PMB151A, and it's here showing you the location of the CFR gene. Strikingly, there was not an absolute identical hit to the PMB151A in any database, although it did show similarities or high identities to other plasmids that circulate in staph, in the genus staph. So these are indicated to you with these lines that you see over here, these bars over here. So there is some similarity to, for example, the PSS01 Coniae, staph Coniae strain, which was isolated in China, and that plasmid also harbored the CFR gene. So the take-home point is the plasmid has some similarity to other known plasmid, but for the most part, it does not seem to have any similarity to other known, complete identity to other described plasmids, and that this plasmid also harbors the CFR gene. Okay. So with the establishment of the reference genome, the stage was set for us to bring in the genome sequences of the other 175 isolates and compare them to this reference genome. And again, I'm not going to walk you through this overly complicated looking bioinformatics pipeline that was established in our lab, but suffice to say, it's a lot of work which basically gets condensed into this. So over hours and hours of computational, of server use, this is the outcome that we are looking for, which is reconstructing a phylogeny of 175 isolates, which is what this looks like. Now, for those students, again, in the audience who are not familiar with molecular phylogeny, a molecular phylogeny is essentially a 2-dimensional path graph, which has 2 components, time and accumulation of nucleotide change. So you can think of that as a graph on an X and Y axis, where Y axis is time and the x-axis is amount of change in nucleotides or amino acids. And what this path graph shows is essentially ancestor-descendant relationships. So a couple of things are very important. So here, as in our data set, as we have found, majority of our isolates were actually SD5, okay, which are shown to you here. And all these red dots that you see are SD5 isolates. And we also saw several, not as many as SD5, but several SD2 isolates. Again, a picture that contrasts with the global distribution of Staph epidermidis, although the caveat being that there may be some sampling bias. 176 is a large number for a genomic study, but really not a very large number for an epidemiological study looking at genotype distribution. Of note is these arrows. If you see over here, now the arrows, wherever they occur, for example, here, for example, here, indicate the strain is linezolid resistant. And it's very striking, and I hope everybody is able to easily see it, that if you do a quick scan of this phylogeny, you would see that majority of the arrows are restricted to the SD5 lineage, which in itself is discernible into two sublineages, which we now call as SD5R for resistant and SD5S for linezolid susceptible. So you do have one odd strain that appears in the SD5S lineage. You also have two SD6 lineage strains, which are very closely related, which basically differ from SD5 in one single nucleotide polymorphism at a single locus. So the take-home point here is that the majority of linezolid resistance, even on genomic analysis, is confirmed to be confined to the SD5, and within SD5 we can recognize two different lineages. So here is a zoom-in view of this area that we see over here, where the separation of SD5 into SD5R and SD5S is somewhat difficult to perceive. So we zoomed in and we made a phylogeny where we included as only the SD5 lineage strains. And what we find is that the couple of things. One is that if you look at this locus, if you look at this section of the tree, you would notice that the branches are very, very small. If you compare it with branches in the lineage over here, which corresponds to SD5S, the branches are very, very long. In fact, some branches are really, really long. So as I mentioned, if you think of a phylogeny essentially as a two-dimensional path graph with change on the x-axis and time on the y-axis, you can infer that there's not much diversity that has been accumulated over time in the SD5R lineage, whereas the susceptible SD5S lineage has accumulated a lot of differences. This is consistent with the idea that the average number of nucleotide differences that we saw in the SD5R lineage was just nearly two single nucleotide polymorphism, or two SNPs, whereas the average number of SNPs that separated SD5R from SD5S were nearly 200 lineages, 200. So the compactness of this cluster then leads us to the hypothesis that either this resistance occurred at the same time, right? And there has not been enough time for the SD5R strains to diversify further. Or second is that the resistance was acquired at one given point and it quickly exploded through the population before they had had time to diversify. So both hypotheses seemed feasible, but we tend to favor the hypothesis that given the tightness of this cluster, the emergence of linezolid resistance, or the spread of linezolid resistance, possibly involves multiple transfers to patients on multiple occasions rather than arising independently. The second point that we notice is that the CFR gene, which is present on plasmid PMB151. So here, if we zoom in again, In black, we have strains that are linazolid resistant. So all strains here are linazolid resistant. Almost all strains have the PMB151 plasmid, except the strain here and the strain here. And almost every strain that has a plasmid has a CFR, except some, like here, 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 and here, that don't have the CFR gene, and also, but are still linazolid resistant. So in other words, the CFR is always identified in strains with PMB151A, but the ST5R strains with PMB151A don't always harbor the CFR, the CFR gene. So next, what we wanted to do was examine the drivers, the mechanisms of why that was the case. And for that purpose, we basically compared the PMB151 CFR containing plasmid with a couple of strains, which I'm just showing you examples here of two strains. We compared all 176. And so here in this graph, the red region basically is the CFR gene. And these blue lines that you see over here are the sequences generated by next generation sequencing runs, and then mapped onto or aligned to the reference CFR containing plasmid. So if another strain has a CFR plasmid, we will see something that would look like this, where you have complete coverage of the CFR region, of the plasmid region with this blue shaded area. So in the middle, what you see is a strain that has the plasmid, but you don't see anything mapping in areas corresponding to the CFR genes. Now, strikingly, what we see is that areas where the CFR gene is not present, but the plasmid, the other components of plasmid are present, we see rearrangements. So these rearrangements are likely mediated by the presence of IS6 family or IS256 family transposases. And a third scenario is where the strain completely lacks the CFR plasmid, in which case, when you take the sequencing reads and try to map them back to the CFR plasmid, you would see a picture that would be like this, where you see spotty sequences aligning to the reference plasmid, but for vast areas, you don't see any sequence aligning to that. The take-home message from this analysis that we confirmed was that the CFR region can often excise from the plasmid via rearrangements likely mediated by IS6 or IS256 gene family. Now, I mentioned earlier that having a plasmid that is bearing a antibiotic resistance gene like the CFR gene creates the scenario where you have a case where the plasmid can rapidly transfer from one isolate to the other. Now, if for the purpose of our hypothesis, if linazolid resistance arose once via acquisition of the CFR plasmid and then rapidly spread through the population, giving rise to the ST5R lineage, then one would hope that the CFR plasmid is mobilizable or can undergo conjugation. Now, when we looked at the data on the conjugation requires a conjugation machinery, which has several different genes. And to keep the long story short, we only found some genes that are involved in the formation of a conjugation machinery, but not, and several other genes were missing. And that gave rise to the hypothesis that the PMB151A plasmid would not be able to transfer from one clone to another clone or one cell to another clone. So we actually did transconjugation assays with representative S. epidermidis strains as donors and S. aureus strains as recipients, and none of those transconjugation assays were successful, which suggested that CFR dissemination by horizontal transfer was very unlikely and would be consistent, which would then be consistent with the limited presence of CFR in the highly clonal ST5R lineage. So that then told us that the resistance didn't arise once, and that bolsters the hypothesis that strain transmission from patient to patient was more likely than rapid spread, or for that measure, linezolid exposure. So in strains which lacked CFR but demonstrated ST5R strains that lacked CFR but demonstrated multi-drug resistance, we looked at other mutational mechanisms, and we found that many of those strains had mutations in the 23S gene, as well as the L3 and L4 proteins, or genes of L3 and L4 proteins in the population. So those single nucleotide mutations would explain why linezolid resistance was pervasive at high levels. Of note, all ST5R strains that were linezolid resistance were also resistant to all the other antibiotics that we assayed, including levofloxacin and others. So the notion that the CFR plasmid is not mobilizable and the ST5R cluster is tightly linked, doesn't show much diversity, suggested that it's patient to patient transmission rather than exposure to linezolid that's leading to adaptation, that's driving adaptation in otherwise susceptible epidermidis strains. That gave rise to the last question that we addressed in this study, and that was that could linezolid exposure lead to selective proliferation of S. epidermidis in the gut microbiota. So here we basically analyze, we determine the microbiome of the patients, which for students who may not have, background on that, we basically take the feces or the stool from patients and we took it on 30th day, 60th day, and the 90th day following linezolid exposure every week. And then amplify the 16, extracted the community DNA, and then amplify the 16S gene, and then do deep sequencing using next generation sequencing techniques methods. And what you see over here is the days of linezolid use in preceding 90 days. So remember this, we're sampling this every week. And here's a distribution of linezolid sensitive and linezolid resistant staphylococci. And what we found basically is that the cumulative exposure to linezolid in the preceding 90 days was significantly higher in patients that actually had bacteremia with linezolid resistant isolates when you compare them to those that had a linezolid sensitive strain in their blood. And that the median duration of linezolid use in patients with staphylococcal emergence was again significantly higher, okay, when compared to patients who did not have any staphylococcal emergence in their stool sample. And then we verified that using two different methods where we looked at the emergence of resistant staphylococci or domination of staphylococci in the gut flora by two ways. One, we mapped the 16S ribosomal RNA sequences to staphylococcal reference genome, which is shown to you here in red. So we don't see any resistance when chemotherapy is initiated for the first 10 days in this particular case. And then we see a spike in the number of sequences of 16S ribosomal RNA sequences that correspond to the 16S gene of Staphylococcus genus. And that continues to increase till it reaches a peak. And then once linezolid is withdrawn, the number of 16S ribosomal RNA sequences falls. Now, these are genus level determinations in the microbiome because we don't have primers in 16S amplification that would allow us to definitively distinguish between the different species in the Staphylococcus genus. So the next thing we did was we also assayed for the amount of CFR relative to 16S ribosomal RNA, and that also showed that virtually mimicked the appearance of the 16S genes. So that basically suggested to us that patients with staphylococcal emergence are associated with linezolid therapy received linezolid prior to having staphylococcal exposure. So that allows us to then say, okay, so while linezolid exposure is not leading for the staph strains to adapt in real time because of exposure to linezolid use, however, is selecting for or selecting for the domination of linezolid resistant staphylococci in patient microbiota. And we looked at several different patients, and most of them showed the similar profiles. And on here we have a patient who had no exposure to linezolid and basically had very little NGS reads corresponding to to very few NGS reads corresponding to the Staphylococcus genus. So in other words, we can conclude that CFR containing Staphylococcus aureus can dominate intestinal microbiota following the linezolid therapy. So to quickly conclude, what we determined in this study was that combinations of significant increase in linezolid use along with transmission of linezolid resistant S. epidermidis clone results in high rates of linezolid resistance among invasive S. epidermidis in leukemia patients. And linezolid use seems to play an important role in dissemination of resistant strains, as was determined by our microbiome and antibiotic use data, by selectively allowing, by selecting for S. epidermidis strains in the microbiota at the cost of other species. And we found a novel clone, the ST5 clone, that caused majority of our infections. And the novelty of that clone was that it's caused numerous invasive infections over the past several years, and that it carried an undescribed plasmid that has significant homology to other staphylococcal plasmids. Fortunately, the plasmid is not transmissible, is not mobilizable from our studies. But the ability of ST5R clone to cause numerous invasive infections in a limited period of time over the past several years suggests that it could either be present in or spread to other healthcare settings, which leads us to suggest that a combination of antibiotic stewardship, which basically means preserving antibiotics, the usage of by precise usage of antibiotics, plus infection control measures to prevent transmission of strains in a nosocomial or a hospital-based setting may help control the spread of this multi-drug resistant pathobiont. So lots of people participated in this study, and I will single out my graduate student, Shichi Lee, who did a bulk of this work, and this bulk of this work was achieved in just a year and a half that Shichi took to just complete this work. So, and other members of the team have also collaborated. Great thanks to Sam Shelban, who's now been a continuing collaborator for over the last 7 years, and we continue to collaborate on various different aspects, and Cesar and Jessica, who contributed the microbiome data. Much of the project was supported by the MD Anderson Cancer Fund and the Ahern Foundation, the Secret Cure Foundation, and NIH grants to Sam as well as Cesar. So with that, I'd be happy to take any questions.
It was a very, very wonderful talk, amazing work on studying the emergence of linezolid drug resistance in Staphylococcus epidermidis. I want to ask you one question, though it is I'm little diverting from Staphylococcus, but this drug is a second-line drug for TB treatment. So if so, does it also going to cause in future, or I'm not sure how susceptible leukemia patients become to develop TB?
And that's a great question. So in TB, first, in TB, most of the drug resistance that we see is mutational, it's chromosomal mutations, and not acquisition or as for by plasmids, because TB is not naturally transformable. So TB eventually acquires resistance to almost every drug that we are using, including fluoroquinolone. So second tier drug eventually is, the mutations might arise in the 23S peptidyl transfer center, and they can compromise linezolid. So I haven't seen much literature on linezolid resistance in tuberculosis, but I wouldn't be surprised if it does arise.
Yeah. Dr. Dash, there is one question, very basic, but yes, very interesting question. Is there any preliminary parameters for antibacterial activity against drug-resistant bacteria? A student has asked this question.
Yes, preliminary parameters for antibacterial activity. I'm not sure I'm fully understanding the question. What sort of preliminary parameters are you looking for antibacterial activity? I'm not sure I fully understand the question. I'm sorry.
That's okay. There is another question. Is there any intermediate metabolite of this drug, linezolid, to which this bacteria is resistant to?
Actually, I don't know that. I don't know an intermediate metabolite for linezolid to which the bacteria might be resistant to.
Okay. There is one more question, Dr. Dash. Would you suggest a modern and efficient method for study of drug resistance plasmids?
Yes, absolutely. Depending upon resources, of course, right? I would. So we've reached, when I was doing my postdoc and I came up with the method of multi-locus sequence typing for streptococci. We used to individually amplify each locus and then sequence each locus from hundreds, thousands of isolates using Sanger sequencing or just dideoxy chain termination sequencing. Now what we find is that it's actually much cheaper to do next generation sequencing.
Sequencing, yes.
Right, rather than Sanger sequencing. So if you have a large collection of isolates, it's not. It's quite easy now to sequence the genome, the complete genome, and then look for plasmids by de novo assembly. So yeah, that would be the modern method of doing that.
Yeah. There is a very basic, but yes, it's being asked by a student that why are bacteria becoming resistant day by day? But that is a matter of concern actually. And this is what you have been working on.
Yes, so there's several causes of why bacteria are becoming resistant. Why resistance to antibiotics is increasing. One is, and especially relevant to what happens back in countries like India, where antibiotics are available without prescription. So you have sore throat, you go to the chemist and you pick up penicillin and erythromycin, and normally there is a course to follow. You have a course to follow, let's say a 7 or a 14-day course, but how many of us do that once the symptoms improve? We don't do it, right? So once the sore throat is gone, we stop taking penicillin. And that process of not completing through the cycle of the entire duration of the antibiotic use results in selection of strains that may be tolerant to that antibiotic, and over a period of time with repeated exposure eventually become resistant to those antibiotics. So one is and this falls squarely into the central point of antibiotic stewardship, right? So doctors are prescribing antibiotics before antibiotic profile is known. That's number one. And so if you don't have good empirical data for your local population saying that, yes, fluoroquinolones will work, in my patients who have this kind of UTI, then you're basically shooting in the blind. And then the patient is not compliant, taking the entire, so you're getting antibiotic resistance from there. The third cause factor for emergence of resistance is the use of antibiotics in the meat industry, in poultry as well as in meat.
A lot.
So what a lot of people don't know is that what a lot of people don't know is that the meat industry uses antibiotics not to keep animals free of infection, but because antibiotic usage results in weight gain. Right? So they're pumping animals with antibiotics to increase the muscle mass of the animals, and eventually animals poop, antibiotic poop goes into the soil and finds ways into the river bodies, and from there the resistance spreads. So these are some of the key reasons why antibiotic resistance continues to increase.
There is one more question. How to make a drug-resistant cell line? Do you know that if there is cisplatin drug resistance colon cancer cell line or not?
I'm sure there is an answer to that question, but I wouldn't know that. I'm a biologist. I don't really work on cancer, so I wouldn't take a guess there.
There is one more last question, Dr. Dash. I'm really sorry keeping you engaged.
No, that's all right.
Yeah.
It's early morning for me. It's night for you, evening for you guys.
Definitely. Is there a database for cystic fibrosis disease related gene? Because CF, Alia Khan has written CF. So I'm presuming it is cystic fibrosis. I'm not sure.
Yes, there is a database for that I can tell you. And database is accessible to everyone. So if you go to the NCBI, which is National Center for Biotechnology and Information website, there is a database called as Database of Single Nucleotide Polymorphisms, DBSNP. And there is actually a variation portal on the NCBI website that you can access, which has all the different point mutations, deletions, etc., that are associated with cystic fibrosis.
Thank you very much, Dr. Dash. It was really an enlightening talk. And you so lucidly, in a lucid manner, you explained to us that how drug resistance is emerging and how are you studying it through various molecular tools like multi-locus sequence typing, then molecular phylogeny path study, and definitely this is going to pave way in future, and we would be able to tackle the global problem of drug resistance, which is emerging for almost all antibiotics available on Earth.
Thank you very much. And if anybody has any questions later on, feel free to reach out to me via email. I'll be most happy to answer if I can.
Thank you. And thank you, Manisha ma'am. It was the whole conference is very, very informative. Students, you should feel lucky and thank the organizers for organizing such wonderful conference with such relevant topics and great speakers. You have brought to this platform for all of us. Thank you so much.
Thank you, Monica. Thank you so much for coming, and thank you, Avdesh. It was a pleasure, and it was such an informative talk. And but we really have to learn so much to understand such difficult things. And because it's a very important, I think it's a very important aspect to be addressed now, because antibiotic resistance. So we've gone to 5th generation cephalosporins, I think, and whatnot. We are on to many more things. So I think it is a very, very important, and maybe you become a Nobel laureate discovering something good, because I know you're in the line You are in the line. I
No,
have got
no,
to
no,
know
no,
many
no,
things
no.
about you. Yes,
No.
you are a big man. So
I'm a
I'm
very,
hoping.
very, very ordinary scientist.
No,
I'm
no,
more an
no.
educator.
I know that. I'm really hopeful, and one day I'll meet you as a Nobel laureate. So thank you. This is your token of appreciation, Avdesh. It's a big honor for us to have you here on
Thank
this
you very much.
platform, and coming to Deshbandhu and giving a talk. It means a lot.
It's
And
as I mentioned, it's a privilege for me because I did graduate from there. I had a good time when I was there.
Yes.
I learned a lot from my teachers, and
Yes.
I think they set a very good foundation.
Thank you. Yes, exactly. Our
It's-
teachers are
it was
really,
excellent.
yes, your teachers were very good, I know, because our old colleagues who retired were really very good. Still, the teachers are very good. So thank you,
Manisha ma'am,
I hope they're better now.
I would like to say one thing very important, that you are an inspiration, sir. You are an inspiration to all our students. And I'm sure after listening to your wonderful talk, they would be quite interested in taking sciences and research in as their careers. So I'm really thankful to you.
Yeah, that could be a
Thank you.
wonderful outcome.
Avdesh, one more thing I would like to say here. We have now Professor Kamal Kumar Gupta, sir. He is the Vice Principal of our college, and he would love to say a few words to you. 1 minute, sir. Let me unmute you. Yeah, just a minute. Please unmute Kamal, sir. So he was very excited when I told him that you are coming. So, वो तो बहुत बातें करते हैं. हमारे old teachers would, just speak lots and lots of good things about Avdesh. He was a very bright student. मैंने सोचा कि अब sir दिख रहे हैं तो sir से बोलवा देते हैं, like how our senior teachers used to feel about you. So, sir, please.
Am I audible? Do you hear me?
Yes, sir.
Hi, Avdesh. And in fact, I'm feeling so happy and so proud to see you here. Of course, I never taught you. I joined the year you have passed. But I was just chatting with Dr. Ravikant. Dr. Ravikant used to remember you so much that every time some brilliant students, he used to say, Avdesh, Talia, and Shivendra. The two students, I don't know if you remember Shivendra. So every time, and there used to be so much of discussions. He always, right now I was also chatting him that your Avdesh is there on the screen. So he immediately joined, and he just look, he could make out. And it's really proud for us to see you here, and listening for the students, really great movements for the teacher. I'm very, very happy. I'm so happy. I'm so proud. Thank you. And of course, your work is really speaking the height you have reached. It's really a proud moment for Deshbandhu fraternity. Thank you.
Absolutely.
Thank you. And we know that you are a very, very busy person. You have a lot of commitment, lot of research work, what is being seen in this platform. But thank you for accepting our invitation. Initially it was Dash, so I couldn't make out. But when Manisha said Avdesh Kalia, I went, oh, he's our student that way. And so thank you for part of this
Avdesh,
conference.
सर तो बता ही रहे हैं. अभी अगर सर होते हमारे सर है रविकांत सर.
I don't know if he's there, but right now maybe all the teachers have retired of your time, because I'm the senior most and I've not taught you. I joined in 91 July. By the time you already left the college.
Yes. I know we're getting all of us are getting old now.
Yes.
And Avdesh, sorry,
that is off the record talks, nothing to do with the conference. So we are back to the college days. It's really great. It's really, thank you. Thank you, Manisha, for having an interaction with our all students. Thank you. But once a student, all-time a student, because I'm also a student of Deshbandhu College, and I always remember that I'm a student. So Deshbandhu students really do wonders.
They really do very well. I know.
And Avdesh, if possible, because I'm looking for the alumni association of Deshbandhu College also, and if possible, sometime we are going to have alumni meet right now in the hybrid on the online. So I'll send an invitation, and if possible, please join for some time.
Yes, absolutely. I'll be honored.
Oh, thank you.
Well,
And
thank you all.
Avdesh,
Thank you very much.
we have one more. I'll just take a minute more. We have SM Jha sir from History Department. He was so thrilled that Avdesh is coming, and sir also tries to just listen to whatever talks, scientific talks are there. He is an ardent biology lover, I must say. He will bless you in just a line. Sir, आप unmute हो गए हैं, sir.
Congratulations, Avdesh. I'm very, very happy seeing you here. I am very, very happy that our students are doing so well in research, and just want to have produced, gem like you. Congratulations and thank you. Very good.
Thank you very much. Thank you.
Thank you, Avdesh. Thank you so much for coming. And please, you are going to be off to your work. I know it would be a very busy day for you now. Thanks a ton. All the blessings from our sides. From all the teachers still in Deshbandhu and your retired teachers, I am conveying blessings to you. Thank you.
Thank you. Thank you again for inviting me. Have a great evening.
Much. God bless you. So we are over with the sessions for today. We are over with the sessions for today. Rahul is here still. Moses is still here. So we are at work, and then we meet tomorrow Sunil sir is also here. So we meet tomorrow at 9 o'clock with a very, very, informative and a very, very big personality from Johns Hopkins University. So please do join, and it is going to be a wonderful talk. She's a very eminent personality. So please do join at 9 o'clock, all of you.
Manisha, there's no need to say big personalities, because it has already been proved for last 2 days. We are meeting such wonderful speakers and big personalities globally, not in context to India or anything. Globally, they are big names. And the research of theirs is speaking the level of what they are doing.
Exactly.
And all my appreciation, congratulations, Manisha. You have really done a great job. And it's matter of proud for zoology department, and not only zoology department, Deshbandhu College and Delhi University.
We learned from you only, sir. Actually, you were the person to start the conferences and national conferences. We पहले start किया, फिर आपने फिर international किया. Because आप लोग रोल मॉडल्स हैं, सुनील सर ने किया।
कई बार बड़ी अच्छी मन से वो कॉम्प्लीमेंट देते हैं, तो ले लेना चाहिए दूसरे
नहीं सर, सच में सर, अगर आप लोग नहीं सिखाते तो हम कहाँ से करते, बताइए आप लोग नहीं सिखाया, बिकॉज जो फाउंडेशंस आप लोग रखते हो हम उसको फॉलो करते हैं।
I can say we started inertia and you take acceleration that way.
Yes, sir. No doubt about it, sir.
We start with the first gear, and now it is moving in the 5th or 6th gear. The car is moving in the 5th.
सर, आपने फाउंडेशन स्टोन ले किया, हमारी ड्यूटी बनती है कि हम इसको धीरे धीरे अच्छा ऊपर ले जाए, हमारे बाद भी जो आए वो और अच्छा करें ना सर।
The future is safe. Future is safe.
थैंक यू सर, आप लोग शाम में आके ज्वाइन किए, आप यहाँ पर थे शाम तक भी। थैंक यू सुनील सर, थैंक यू सो मच।
God bless you. And at the same time, I can see our dear Rahul also. So future ka bhi future.
Moses,
I'm looking a very bright future, and Moses is also there.
Yes, Moses, switch on your video.
Yeah, he is. So this is really a proud moment for us, and we are just basically a chain what our seniors have told, and we are carrying it further. And now,
very true, sir. अच्छा, हमारे बच्चे भी आगे अच्छा सीखें, हमारे स्टूडेंट्स भी बहुत अच्छा सीखें, और आगे हमारे जो आने वाली यंग फैकल्टी है वो भी बहुत अच्छा करे।
And another thing is, I was just chatting with Dr. Ravikant about him. So he was remembering everything, and yes, and then I asked him, I think he has passed in 91, because I joined in the July. Yes, I remember very well. Otherwise, Dr. Ravikant remember years. It's not that easy, but he was remembering. Yes, I was there. And Ranjana has joined the same year in the month of January. So Ranjana must have taught for a couple of months, that's all. But I joined the next academic year.
He was intelligent, sir, because वो MSc में था हमारे साथ, तो अच्छा था, और क्योंकि उसका बाद में PhD भी एम्स से चला गया था, एम्स में PhD करने के लिए, अच्छा स्टूडेंट था ही, बिकॉज एमएससी में जब हम मिले थे तो वो अच्छा काफी अच्छा था. He was a very bright person. So कर रहे हैं बच्चे अच्छा. अभी हमारे ये वाले आने वाले स्टूडेंट्स जिनको हम पढ़ा रहे हैं. थैंक यू सुनील सर. सुनील सर को अनम्यूट कर दो जरा. सर आप अनम्यूट हैं सर, एक सेकेंड, क्योंकि काफी. हाँ सर अनम्यूट करिए अपने आप को.
Okay, congratulations to all the organizing
Thank you,
team
sir.
and the technical support जो काम कर रही है. Definitely, it's a wonderful task, wonderful job. And I think please check it. Some students are there or not. If they are not, you can just ask them to leave, and then we can have our offline meeting or sorry background meeting. Just see कोई बच्चा है, they can leave.
So students, you are there in the classroom right now, you can leave. We'll start the session tomorrow early in the morning.
We have some we will conduct a meeting to conduct the tomorrow's program. So those who are in the meeting, they can leave now.
There are more than 50 students are there, sir, still.
सर घर जा हम या तो हम उस पे कर ले, घर पे जाके गूगल मीट
नहीं, hardly 10 minutes. 10 minutes. Just wait for few seconds. Rahul sir. I have a proposal. I am trying to produce one movie. So we can cast.
Cancer se movie, my God.
Very good. Excellent job. Shahzad, Rahul, and then this.
The girls left
Sushma,
थोड़ी देर पहले. हाँ ये दोनों गौरी सुषमा they left अभी थोड़ी देर पहले ही, बस बिकॉज बहुत रात हो जाती इन लोगों को, बच्चों दोनों लड़कियों को, तो इनको लीव करवा दिया हमने बोला अब आप जाओ.
ठीक है it's good.
तो दोनों अभी
दादाजी का भी कोई रोल है मूवी के अंदर.
Still 42. Those students who are in the meeting, they can leave now. We have another meeting.
हम लोग कल का वैलिडिक्टरी का हम डिसाइड कर रहे हैं बच्चे.
हाँ.
कल सर चीफ गेस्ट होंगे और मैंने कुछ वो किया था सर, एक तो वो करना है.
Rahul?
Yes, sir.
I think we heard some technical problems. Some students were creating trouble. Could you find out what was that?
No, sir, ऐसा लग रहा था कि वो कोई बाहर से आके किसी ने ज्वाइन कर लिया था, बिकॉज़ ओपन एक्सेस था ना इसका.
Okay. सर एक्चुअली सर
कोई भी ज्वाइन कर सकता था, तो वो ही हुआ था, बट उसके बाद सब ठीक हो गया. Now everybody is joining. पता नहीं वो गलती से आया या क्या हुआ समझ नहीं आया, बट उसने 5-10 मिनट तो आया रहा. मेरे ख्याल में 10 मिनट तो रहा ना वो. 10 मिनट आता रहा.
10-15 मिनट था मैम.
एक्चुअली सर सिक्योरिटी में हम अगर ये लॉक मीटिंग कर देते तो सर फिर दूसरे पार्टिसिपेंट्स आ नहीं सकते थे. तो इसलिए हमें अनेबल करना पड़ा. नेक्स्ट टाइम हम क्या कर सकते हैं, सभी पार्टिसिपेंट्स को पहले ही बुला सकते हैं अंदर, या मैनुअली कर सकते हैं. सर पार्टिसिपेंट ज्यादा थे तो हम मैनुअली नहीं कर सकते थे. तो नेक्स्ट टाइम हम क्या कर सकते हैं, मैनुअली इंसर्ट कर सकते हैं.
Otherwise it was हाँ वो थोड़ा सा इशू रहा, but it went very well. राज की प्रेजेंटेशन भी अच्छी रही, बच्चों ने अच्छा बोला. I heard every presentation. It was good. स्पीकर बड़े अच्छे मैम ने अरेंज किए हैं, है ना. बहुत ही एक्सीलेंट स्पीकर.
Yes, sir.
And Sunil, I want to give full compliment to Rahul also. Rahul is really a part of this family. The way he's working, I'm really impressed.
Yes.
I'm very happy.
I'm also happy. Seriously, the way he's conducting,
he's really committed to the work. And for last 2 days, I visit very often to the maths lab. and it wouldn't look that he has joined recently.
Yeah, I won't
Seems like
look
it's a part.
that he joined right now. It
हाँ.
seems that he's now colleague since
Yeah,
decades.
और अभी तो इसको officially वो class कोई लिया नहीं है, but
Thank you so much, sir.
अच्छा, एक और बात सर, इसको कभी भी आवाज लगाओ तो इतना engrossed होता है ना, इसको वहां जाके बोलना पड़ता है, राहुल इतना engrossed होता है सुनने में कि बिल्कुल ही मुश्किल हो जाता है यहाँ से आवाज लगाना, जबकि पीछे ही बैठा हुआ है, इतना engrossed. यस मैम घबरा के ना फिर एकदम यस मैम हटा के अपने ईयरफोन. तो अच्छी बात है, अच्छी से काम करना अच्छा होता है, आगे बढ़ना है, इन लोग को अच्छा करना है. सर अब हम आप लोगों ने सिखाया, हम सीखे, अब हम सिखा रहे हैं, अब ये सीखें, ये जितने भी हमारे बच्चे हैं सारे ये सीखें.
मनीषा हम अभी भी सिखा सकते हैं, ऐसी बात नहीं है. ये नहीं है कि बस तुम ही सिखाओगे.
हम हर रोज सीख रहे हैं. नहीं सर, हम हर रोज सीखते हैं, सच में सीखते हैं, रोज ही सीखते हैं.
अच्छा, एक अभी जो वो क्लिपिंग था ना वीडियो वाला, जिसमें अवधेश और वो कन्वर्सेशन, उसके क्लिप करके ना जूलॉजी डिपार्टमेंट की वेबसाइट पर अपलोड कर देना, है ना. Informal talk with the director of whatever the Anderson Institute, है ना, that way.
कल मॉर्निंग में कितने बजे से स्टार्ट है?
कल सर मॉर्निंग में हमारा है तो 9 बजे स्टार्ट. स्पीकर बहुत धांसू हैं, मतलब मुझे लगा कि जाने के वक्त अच्छी स्पीकर एक हो जाए, तो वो हैं. फिर अंजू श्रीवास्तव मैम का भी है. फिर सर एक सेकेंड सर, बस अंजू श्रीवास्तव मैम का है और सिंथिया का है. अरे सर अभी अडांकी सर का भी है, अडांकी सर का भी है, राइबोसोमल प्रोटीन सिग्नलिंग. ही इज अ वेरी वेरी वेल नोन पर्सनालिटी, वो भी मिस करने लायक नहीं है. और ये 2 टॉक्स तो बहुत ही अच्छे हैं. और सर फिर उसके बाद वो अंजू श्रीवास्तव मैम का 3rd टॉक है. सर हमारे 4 टॉक्स हो जाते हैं एक सेशन में, एक दिन में ना. और
तो इवनिंग में किस, मॉर्निंग में किस किन का है और इवनिंग में
सर सबसे पहले आप लोग सिंथिया जहनाव का जरूर अटेंड करिएगा. देन अडांकी सर का होगा 2nd टॉक 9:40 पे, 9 o'clock पे 1st टॉक, 9:40 पे 2nd टॉक. सर उसके बाद 10:20 पे 3rd टॉक होगा अंजू श्रीवास्तव मैम का.
वो तो मुझे ही करना है
जी सर. फिर सर ओरल प्रेजेंटेशंस है 11:40 से और 1:10 तक.
तो मुझे 11:40 से 1:10 तक आई हैव टू सी.
नहीं सर, आप वो होंगे ना सर, इसमें 10-20 वाले पे होंगे ना आप. अंजू मैम के चेयर है ना आप.
अच्छा ठीक है.
हाँ जी सर, वो रहिएगा आप 10:20 पे अंजू मैम को के सेशन को चेयर करने के लिए. और फिर सर हम लोग का सर एक बहुत ही इम्पोर्टेंट और बड़ा अच्छा सा है, एक्सपीरियंसेस ऑफ कैंसर सर्वाइवर्स. उसमें हमारे 2 टीचर्स को हमने वो किया है, जो डीयू में थे 2 लेडी टीचर्स को हमने रिक्वेस्ट किया था. So they will also be giving their life, story. वो बहुत अच्छा सेशन होना चाहिए.
ठीक है.
That's great.
फिर वैलेडिक्टरी सर हमारा 2:40 पे है, स्टार्ट हो रहा. वही शाम बच जाए, शाम ही हो जाएगी. 2:40 से 1.5 घंटे का तो होगा ही, 1-1.5 घंटे का. तो फर्स्ट 2 टॉक्स भी हैं, फिर 3rd टॉक भी है ही. अच्छे होंगे टॉक सारे.
कल लोग कमल सर आप कल आ रहे हैं?
If you feel I can come to the college and no problem.
Okay, I think we should celebrate tomorrow. कल ही सेलिब्रेट करते हैं वैलेडिक्टरी या फिर मंडे को रख लेते हैं.
मंडे को ही रख लेते हैं सर.
मंडे को रख लेते हैं.
मंडे को पता नहीं कितने लोग आ पाएंगे सर.
ठीक
Monday
है।
को we will invite Rajiv ji also in the celebration and let he also be part of the celebration.
Yes sir.
वैसे हमारे को राजीव सर ने करना चाहिए ट्रीट, कितनी मेहनत कर रहे हैं सारे टीचर्स।
I'll treat. Don't worry. The
हाँ,
vice
it's
आप
not
कैसे
about treat.
करोगे? आप तो हमें हमेशा कर देते हो।
Nee,
आप
Manisha,
सर को पकड़
Manisha,
लो।
not in the capacity of zoology, in the capacity of vice principal, I can treat you. है ना? अभी तक तो आपने in the capacity of faculty या colleague की करा है ना? अब admin की capacity में I can treat you. You need a reason to celebrate and treat.
Yes sir, yes sir.
Okay, fixed.
तो मंडे को
Monday,
मिलते
I'll throw
हैं
a party.
मंडे को मिलते हैं सर। ठीक है सर, फिर अब
Okay.
हम निकल जाते हैं फिर।
हाँ, या please keep it up, है ना?
जी
निकलो।
सर।
Be careful. ध्यान से जाओ।
थैंक यू सर, थैंक यू सो मच।
Okay,
थैंक यू
bye-bye.
सुनील सर, थैंक यू शहजाद,
Bye bye.
थैंक यू राहुल, थैंक यू शहजाद, थैंक यू
Thanks, sir.
सुनील सर।
Bye bye
Then,
everyone. Bye everyone.
Sushma, thank you so much. Thank you everybody. Take care. Kiran, thank you. Gauri, thank you. Gauri, thank you so much. Gauri, Kiran, thank you so much. सब लोग को बहुत सारा thank you. कल मिलते हैं।
Okay,
break के
bye-bye.
बाद।
Take care. God bless you.
Bye, bye everyone. Bye, bye, bye.
Bye
God bless you all.
Shehzad.
Bye, sir.
Bye, ma'am. Bye, sir.
Bye. Take
Good
care.
night.
Take care. Bye bye.
Good night.
Bye.
When should I end the session?
End कर दे बेटा मीटिंग। कर दो बच्चे, हाँ कर दो।